like you said.
So good morning, everybody.
I'll go 20 meters below
This is
you.
Chris Kelly, serving as one of your co-leads on the Okeanos Explorer for dive number 3 of the cruise that's titled La Lima
We can finish up
Okeanos.
white balance.
Sitting next to me is...
And this is Chris Ma from the Smithsonian's National Museum of Natural History. We're beginning our pre-dive meeting just before our descent to the bottom
13 more meters
for this
to go,
unnamed
and
seamount in the
10 meters to go.
region.
And to my left is...
This is Bashkar Malik, one of the physical scientists on board, and welcome here.
So Mashko, you want to walk us through the imagery? Those of you who are should be able to see a map on feed 3, I believe.
Telestream 3 should be showing you a screen showing the seafloor mapping that we were able to conduct last night. And the profile I've drawn on the screen shows the dive track for today's dive. This is the start point. We will be arriving on the seafloor at around a depth of 2,500 meters, and then we will be
Z bias. And manually
climbing up on the seafloor to a depth of about 2,450 meters.
Right.
This total length will be around 750 to 800 meters of dive track.
So, Chris, do you have any pre-dive comments here to make?
Looking down.
Well, I'm really happy to be here for my second Okeanos Explorer dive. I know that
Video
we've
down
been
with the white balance.
We've had a busy Friday morning, and our ROV has been deployed very efficiently and very quickly today. And so we're rapidly approaching the bottom and looking forward to an exciting dive of a seamount, which we've never seen
auto heading
before.
engaged.
Yeah, we don't have a name for this seamount.
My camera
it was
is
a small one that was mapped
out.
by the Okeanos Explorer, most of it. There's
I got you.
a little part that
Titan.
didn't get mapped, but most of it was
Titan.
mapped
All right,
in
Delta 20.
2015.
I'm going to start backing.
When the ship did come down here for 2 cruises.
Copy.
One was mapping,
The other
and the
was
other was
good.
an
Pretty
ROV
tight.
cruise.
Still about to 30.
Even though we had some
Plenty of
imagery
room.
for this
Good
site,
delta.
we
You're
did not
not
dive
auto
on it. This is
heading.
not one of our dive sites in 2015.
I'm
so
happy.
this is brand new. It's a whole new site.
Just you and the edge of
We
fish
don't know
eye.
what we're going to find. We have
Perfect.
no clue, but we're pretty excited. We're in a little bit
Purposes look
deeper
good.
range for some of
Pretty
the
much
very
180 from
high-density
one another.
communities that we've seen elsewhere
Turns to
during the
0.
capstone
All as well.
cruises. So we're very, very interested in
Backing
determining
5-0.
whether 2,500 meters could be sort of a maximum depth for some of these glorious bamboo communities that we've seen in some other sites here. We're currently at 2,571 meters. And so I would love to have that current hypothesis dashed to pieces and we can land in a really nice community, but you just never know what we're going to find. Mashcore is zooming around here on the seamount to give you an idea of what its topography looks like. I'm pretty sure this is a geo, which means that it's a flat top seamount.
Push back pretty hard.
It's got a little bit of texture on the
6-0
surface.
now.
So
And
you're
a
going
flat
to come
top
back and turn to
seamounts are
starboard?
all believed to have been
yes,
at
that's
the
correct.
surface at some point in their history millions
line
of
up with
years
you, and then
ago,
we
and
can,
that's
find
why
bottom.
it's flat. The
Good,
top
see
was
myself in the
sheared
PTZ.
off and or masked
once I get you in
by
Z's, we can,
erosion coupled with the
I'll turn
development of reefs
port you, turn to starboard, we'll
around
meet
it.
northbound.
And so that's why you get this
Sounds good.
beautiful
Okay.
flat top. Right next to it, you can sort of see a seamount that perhaps also was slightly above the
I
surface a
call
little
that
bit
stowed for the shilling.
to a lesser extent, and the seamount that we're
Looks
diving
good.
on
Those
used to
square
have a
remains
sort of a conical summit, but not anymore, and it probably
All right.
lost most of its
Raises
summit
back to you,
probably
hydraulics.
about 80 million years ago.
Here, actually,
Wow, that's
So
the
bring
your
Cretaceous,
drawer in.
I believe.
That's
Yeah,
the
sounds good.
Cretaceous, time of dinosaurs.
As long as we've got our sort of pre-dive meeting
Starting to see myself.
rolling, are there any people in the Okeanos shoreside
Let's see
who
what
have
it's
anything
made
to
HD.
say before
Looks good.
we begin
Okay, I'll
the
bring my
dive
rotator
in
up
a
to
few
90,
minutes?
and then you'll be directly below me. I turn to port, you turn to starboard, meet up northbound.
This is Amanda
Sounds like a plan.
here. I don't know if you wanted me to say
Right,
anything
Tyler's
about
above
the
me.
midwater portion or if you guys
No
have any
danger of
questions
it
on
getting
that.
entangled as
Amanda, give a plug
looks
for
good.
your portion of the dive right now. You have an open mic.
All right.
What was that?
Let's meet northbound.
Give a plug for the
Northbound.
midwater
Sorry.
transects right now. You've got an open
Yeah.
mic.
So, anyone who is listening, we'll be doing something that we don't do on every dive at the end of this dive, where we have an extended dive time. We have a couple extra hours to do some midwater work. So we'll actually, instead of just transiting up through the water column on our way up,
I'll bring my rotator to like 50.
At, a series of different steps and take a slow look around. And we see some really interesting creatures out there, all kinds of different gelatinous animals and
altitude 28
stations
all as well.
Fish we sometimes find. And almost every time that we've done these out in the middle of the Pacific here, we've found either something we don't quite know what it is or something that's never been seen anywhere near here before. So we're really excited to see what
looks good.
Today. Those transects will be from about 800 to 300 meters.
Yeah, there's
we'll be taking a look at what
there's
the
54.
environmental properties
I'm fine coming down here if you are.
As we go down in it,
coming down.
in order to
come down
kind of
15.
guide what
Sure.
depth we can do that work at. So I know Mike Ford will be on the lookout for that information as we start getting ready for the midwater work, so that he can pick our target depth for that.
Great, thank you, Amanda. And those of you who have participated in some of the
midwater
dives
meters,
over the past couple of years here, I'm sure you can attest to the fact that we've seen some rather incredible animals. Some amazing stuff.
I got altitude 38, so
really
it should be
toothy, snarly
Starting to see
fish.
bottom now.
You name it. Eel-like things, beautiful jellies. I can't even remember how many things I've seen in midwater.
The midwater presents an entirely sort of separate group of animals from the ones we see on the bottom. These are the things that swim around and float in the middle of the ocean. And so that'll include, as we've mentioned,
like tethers,
jellies, jelly-like animals, lots of fishes. We see lots of cephalopods that swim in this area. So after the dive concludes later on, for us, that'll be this afternoon, we'll be having about a 2-hour sort of transect through this area to see some really exciting and a very different kind of animal habitat.
This looks great.
All right, folks, as you can see from feed 1, if you switch your computers over to feed 1, we are on the bottom, and we've already got our first organism up there in the upper left, which is a glass sponge.
I'm going to do my CTDs check.
Copy that.
They're going to settle in and do everything they need to do to get us ready to move, but in the meantime, we'll go ahead and describe what we're seeing here.
Yeah, well, actually, just give us 1 moment to set up, and we'll be right with you.
It almost... so it's like a very fine manganese cobbling with sediment in between. It's an interesting texture.
Set up Z-bias. We go to 11.
So I think there is soft sediment underneath these cobbles, which look to me like they're manganese nodules. We'll get a better look. And that sponge in the background there, which we'll undoubtedly get a close-up of a little bit in a few minutes, is something that's, I'm pretty sure is called Semperella. It's in the family Pheranatidae. And many of the species in that genus prefer
I just want to give you a heads up, we are
sediments.
on bottom.
They've got a very nice way of anchoring into the sediment there. You can kind of see they've got a sort of these grappling hooks, these especially modified spicules to actually essentially do a transfer, kind of anchor, sort of like roots.
I heard you guys are already down. Would you mind if we put some wrenches on the nuts for a couple minutes?
Pilots are still there. When they get to the bottom, they've got a bunch of items that they need to take care of, some housekeeping stuff before they're ready to go.
Sounds good. Yeah, I think you got a little bit of time.
As they're settling in.
Sorry, Washley, can you say that again? Might have missed.
We'll be proceeding to explore the bottom area here, but we're also looking for specific types of samples that are of interest to our exploratory research. These are often subjects that are either new species or species that are part of the overwhelming community of organisms that we're studying in the area. We'll do a lot of sample recovery based on feedback from our shoreside scientist team. as well as what we know about the community of organisms that we're studying. Sometimes we'll encounter something
Sorry,
new and
Roland, can you
undescribed.
repeat, please?
All right, co-pilot. Happy with lighting?
Yeah.
Video's happy.
We're currently looking at an
Video's
interesting
happy with lighting.
All right, great.
bottom at about 2,500 meters.
Yeah, I guess so. We could do that
These
for a while.
are
Okay.
manganese nodules. They're precipitated minerals from the seawater that form into unusual round sort of cobble-like structures, and looks like they're covering the sea bottom, and there's a lot of white or to light colored sediment in between them. We have
Copy that.
a very nice glass sponge that we're coming up on.
Yeah, watch leads.
Here, which
We're all
Chris
set up. So
Kelly
whenever you're
identified
ready.
as Semperella. This is a glass sponge. This is an animal, and its body is composed of silicon dioxide or glass. We'll see many of these types of animals as we proceed along the bottom.
Okay, so it looks like we're getting close to our first subject for today's dive.
It's quite a sponge, isn't it?
A closer
Yeah.
look at this does lead me to believe that the genus is Semperella.
How big is that?
It's a glass sponge.
Yeah,
we can
probably
get the
lasers.
lasers on.
Laser's coming on.
It's at least a meter, I think, right?
Laser's on.
So the members
Yeah,
in this
that's
family,
10, yeah.
Pheronematidae,
Easily a meter.
all sort of anchor to the substrate with this loose arrangement of spicules that are modified to support them. And some of these actually grapple onto rocks. That's that sort of brown area you see at the base of this particular sponge. And they'll no doubt zoom in on that so you can get a look at this in a moment. We're just looking at the lasers as they pan across this thing. This looks like it's almost
Okay,
a
whenever
meter
ready,
high.
you want to zoom in.
Yep, go for it.
Quite a
Some
large
creature
sponge.
living at the very top. There's also something coming off to this left
So
side.
there's the mat, and
tilt
there's
down to
more
see the
of
full
these
base.
sort of grappling hook spicules that are sticking down into the sediment that are stabilizing it in here. So there you can
that
actually
the view?
see the
Perfect.
individual threads. These are called spicules. These are produced by the sponge.
Okay,
They're
a slow
brown because
tilt up.
they're dead, but they're still functioning to hold
Slowly
sponge in
tilting
place.
out.
Go pilot deck.
So all of the members of
I got
this
you.
family of
Hey, would you mind
all
rotating
continuous
either way,
Yeah,
whichever way is
continuous
better?
like you're going to a stop when I see a thing poking out of the
You ready
left
for me to
side.
come up?
Copy that.
Yeah, that'd be good.
All right, coming up. Let me know when to stop.
To the bottom
Okay.
of the silicious plate. We are looking at sort of the convex surface of the sponge. On the other side, it's a little more concave.
Hold there.
Keep going.
These are
Hold it.
oscula, these holes on the side of the sponge. And
Right here,
it's
see it?
called a soup plate.
Yeah, we only got two
covering all
more,
of
so we'll
these
call you when we're
Okay.
done.
Continue
Thanks,
from the
holes
top.
to prevent unwanted things from crawling
Alright.
in there.
Coming up.
The
Water,
water
sorry.
actually comes in through these holes. Then the sponge filters. Yeah, that's kind of weird. Chris is pointing out sort of a pimple on the left side.
There's a
And I
volcanic
think
shaped
we're going to come up to a
protrusion
creature on the
that
left
has
now.
kind of an
Creature on the
unusual
left eye.
branching structure emerging from it. So that might be
Possibly
some other organism
snail.
or just an irregularity.
There he
Oh,
is.
there's a spot.
Oh,
Here's a
there's-
snail.
There's a snail, yep.
And tilt over to it and I'll zoom in.
So what we're seeing is a gastropod, an animal, a snail, similar to the type of
Excellent.
animal that you might see in your garden at home, except here at 2,500 meters, it has likely a very different kind of life mode. It might be on the sponge, feeding on it, or it might simply be crawling up it for
Okay, and just go ahead and
protection or
continue to the
some
top and
other
then we'll go
reason.
full screen.
a lot of these
Okay,
snails
co-pilot,
are
we're all done, all
poorly
clear.
understood,
Quick.
and
Thank you.
Thanks, Chris.
we'll probably
Okay,
take a good
And
image
we'll
of it
do
and
full
forward it
screen.
to a specialist
Copy
Light
that.
tilt
for
down.
identification.
Slight tilt down. All right, watch leads. Any other part of this you want another close-up of?
No, thank you very much. I think we're good back here. If video's good.
All right, great. We're just going to go up slope. If you see anything, just shout out, and I'll do my best to get it.
can we make sure
Thank
that
you
the
very much.
lasers
And
cross
who's
over
up
the
there?
laser?
You guys need to introduce yourselves. All we see is the top of your heads.
Ah, that's correct. Sitting front row. We're all from Global Foundation for Ocean Exploration, GFOE. Sitting pilot right now is Sean Kennison. And I'll let everyone else introduce themselves.
Sitting navigation right now is Andy Lister.
Co-pilot Levi Unima.
Video shading shares Emily Narrow.
And way off to the right, you can see nothing but a hand is Roland Bryan.
All right, video, are you happy with this?
Video's clear.
Great. You're up to my port pilot. That was Roland Bryant.
Copy that. I'll come
video
center.
over in the corner, and Emily is right here next to us as well, who's doing the clipping.
And
we have intern Nicola, who's helping us log species into our annotation
according to
database.
HIPAC, but let's use sonars instead.
For those of you who just came on, we'll repeat our names. I'm Chris Kelly.
I'd say start with 30 meters north, see if it changes.
This is Chris Ma. I've forgotten my name. Smithsonian Museum of Natural History. I'm a sea star expert.
Yeah, it's
among other things.
It's pretty easy to forget the Smithsonian Institute.
You guys ready for that move?
Very well known.
Actually, it's an institution, not an
We're set up pretty good. Deck's happy, right?
Yeah, they're done working on the way.
If I forget my name next time, all I have to do is look at your.
We're ready to put our ship move.
I just remember half of that.
30 meters, due north, 0.029 knots.
For those of you who are interested, I also am on Twitter as Ekainoblog. You may know me as a screen grabber from before.
Good copy.
Some of you have noticed my absence.
Very loud.
I would like to comment that true, I'm not screen grabbing. I'm actually
on top of that.
With these dives. So bear with me, and
do we have any?
Next time around, I promise I'll help you out with the screen grabs and so forth.
Like mine? I'm not sure. I think we should have backups, but I'm not.
I'm going to snap on the sediment.
Copy that. Getting closer to it. Co-pilot. Tyler, tell me if I
So we're coming on an
Old
interesting
sponge.
little sediment
Good
patch
copy.
in the midst of this manganese nodule
I give you some more tether
landscape.
pilot.
I'm
That's
pretty
good.
sure
I'll
this
stop
is
there
probably
just in
the dead
case.
portion of one of these Semperella sponges. So the
All right,
sponge
video
is
if you
long
want
gone, maybe
quick
blown
zoom.
off by currents.
And that's what it is.
Yep. All right, video when you're happy.
At least from the sponge's perspective, there was a catastrophic event at some point in the past.
Indeed
right.
so.
Not only did he lose his head, he lost all the rest of them too, except for
Yes.
his roots.
This is the deep sea version of an abandoned house.
Yes.
It's very sad,
We have a minute before
but
the ship
in a
starts moving. There might be
sad in a
something.
happy way.
Yeah, I'm
sort of.
panning over that way.
Yeah, okay.
Just trying to maintain radius around you.
Yeah.
I heard somebody chime in. Who we got on the line right now?
Just want to back up and turn. Maybe some right there, right?
Yeah, I'm coming up just to get out of your way.
So,
Copy, I won't
the
come
name
too
of our
far
expedition is the La
Is there
Lima
sediment buildup? I
Okamoana,
see it's-
which roughly
Those are
translates
from
to
my,
Partnership for
yeah.
the Ocean. And
Must be some
nothing
sediment
could
on the
be
bottom.
truer
Yeah.
because we actually have on shore a number of scientists who are contributing their talents
This
in
looks like the same
assisting
thing, right,
to help
video?
us identify the many animals and organisms
it
that
is.
we see
Copy that.
here at this deep sea Guyot. So, and as well as the pilots and video technicians and other people that are helping us. Science is a cooperative effort, and we work hard to live up to the cooperation theme that the title of our expedition
Do a quick turn.
suggests.
I thought it was just that we
20.
needed a lot of help, rather than just pure collaborating.
It's going
Well,
down.
yes.
We need lots, yeah.
Yeah,
that's
quick
it.
zoom.
This is-
Probably
But,
going to go
collaboration as opposed to
Yeah,
That's
no,
true.
just my telling all of you what to do, which would be kind of fun, but, monotonous.
So as they're zooming
video.
in on this,
Yeah.
you can see the substrate here, and these are manganese nodules, and these are going to be targeted at some point in the future for deep sea mining, because these nodules actually are packed with all types of commercially valuable minerals, such as cobalt and
Too small.
titanium,
Very
and,
small.
Yeah.
All right, we'll leave.
iron.
Have you guys
and
scrubbed the
copper
ground to
and
see if those
tellurium.
nodules
and
move?
tellurium.
I
So
can go
all
back to
types
where I
of
sat.
things. So
I
we
definitely
actually
leave marks.
did land in a little bit of a nodule
Tips
bed
interest to
on
those
the
guys.
ridge because there's a
Yeah.
little flat part
That's
of the
good to know
ridge.
what we're sitting on.
And we're going to transect through this bed, and then we'll be at the harder, more
Yeah, I can see
rugged
Sirius
terrain
B coming
in
up
a
to a
short
couple
time
lines, which is probably in
here.
front
But
of your
it's kind of
skids.
interesting
Yeah.
to see this transition.
Is that what
There's
those are?
not a whole
Yeah.
lot of larger animals
Yeah.
that we can video
There's a
easily
nice mark.
that live in these nodule
But it doesn't kick up
beds.
any sediment, which is
However,
nice.
there are studies in a area called the Clarion-Clipperton Fracture Zone to the southeast of Hawaii, where if you actually look closely, you can see all types of small animals that are living in there, and quite a thriving
Yeah,
community. Yeah.
watch these. If you want to see where we sat,
we zoom into those.
Yeah.
Yeah, so this clearly shows that underneath these nodules, it's not a hard substrate, it's sediment, and this is a nodule formation. So deep sea mining is, if they eventually wind up developing the technology to do this, most likely they would probably use some type of a suction device to get
just
these
went
nodules
off screen
up
left in your
because
skid.
they are loose on the surface, as you can see. They're easily
Copy that.
displaced
We'll
by
hit this in,
the.
and we can look for it.
It's there in your image.
By the
very small, white.
ROV.
Yeah.
When it moved around.
Urchin.
Oh yeah, look at that. You've got a little animal. This is what I'm talking about. If you look closely, you can actually see some things. It looks like either a small sponge or an urchin. It could be either one. That sort of white thing on the nodule.
Yeah, it's
Yeah, and Scott France actually
And
concurred.
pulling
There's
out full of
probably a
eye.
lot of really interesting microfauna living in and amongst this bed.
Run lasers across.
It's just a different community
See how small
than
it
you
really
see,
is.
On the rockier terrain that we'll encounter further upslope.
Wouldn't even guess.
Yes,
Laser
and that's a good point. Even though it appears
centimeter,
relatively barren, there's probably a great deal of life that you see in and amongst these nodules, and even at smaller scales, smaller microbial forms, as well as a multitude of micro invertebrates, worms, snails, sea urchins, and other things.
For our purposes,
there's very we don't know very much about the life in this kind of setting, and so that's what we're here to do, is try to understand what's here and what kind of ecology might be present.
Yes, feel free.
So while we're traversing over this
what makes what?
area right here, we just want to let you know who all is in the event log right now. We've got Jessica Robinson, Amy Bowman, Andrew O'Brien, Asako Matsumoto, of course, the two of us, Derek Sutcliffe, John Smith, Les Watling, and we've got Nikolai Rodriguez, who's sitting next to us as well.
I don't see anything.
Steve Auschwitz. Hello,
another
Steve.
sponge in the background, top right.
So we got a bunch of institutions and countries actually represented here. So Osako is in Japan.
All right, great.
And then we've got a bunch of U.S. institutions
right?
here. We've got the University of Hawaii with Les Watling and John Smith. Nicola is with Hilo, University of Hawaii Hilo. And Scott France is with the University of Louisiana, Lafayette. And
I see that sponge video. I think we're too far from it right now, though.
Sorry, Steve.
The Cortez Lab.
Cortez Lab, Cortez Lab.
It's a couple of
and Timothy Shank is joining us from the Woods Hole Oceanographic Institute in Massachusetts. Jessica Robinson, I'm not absolutely positive where you are contacting us from. Scott let me know that Steve is with Temple University. Sorry, Steve, and thanks, Scott. Senior moment. I will have a few of them on every dive. So anyway, thank you, all of you in the event log, for joining us this morning, and we'll get some others, I'm quite sure, as the dive goes on.
I'd say that fix we did yesterday worked. 840K on that's pretty good.
Copy. We turned it off, though. It did not improve.
Yeah, that's
real cam.
Okay, so and also while we are settling in here, we just want to let you know a little bit of the Okeanos sampling priorities. Most of you already know about this already.
But they're each 20 mega ohm.
We basically try to, for every dive, we try to reach a balance of covering a certain amount of distance so we can get an understanding of the community ecology at a particular site. We also like to get plenty of close-ups.
50, right?
Yeah, it was 50 with that.
Yeah.
Swimming shrimp.
So it looks a little bigger.
Well, I'm obviously going to have to leave and go get my Starburst candies, but that was a ship store announcement. I paused for a moment. But anyway, the sampling priorities. So we obviously want to cover some distance. We want to get those close-up images as well as we're getting right now. This looks like it's a Canthophyra, a particular shrimp that sort of drifts over the bottom. that looks like a drunken sailor most of the time. Like
lasers
like
clear.
right now.
But
The other thing is too, for actual specimens, we do like to collect somewhere
Drifting
between
away
2
from
to
you.
3 biologics and 2 to 3 rocks, and keep
That is
it
good if you
at a
wanna follow.
relatively low number compared
Copy
to
and
other
follow for a
types
little
of
bit.
cruises and other ships, because we have so many objectives with so many
I'm gonna let you
different
go off screen.
interests that we're
I got
trying
you, I'm
to
tight.
accommodate here.
Copy that.
And also it's probably
Video
a
whenever
more
you're
interesting
happy. I
dive
don't
for
want
the
to follow
public.
too
Okay.
much
Don't
more.
just
Clear.
sample out the
All
wazoo.
good? Great. I'll let them go out.
But the types of specimens we collect are hopefully new species to science, or new records for the area, or if we get
Alright, Kippa, you get
something
my
that's
picture.
very dominant in the community and we're not sure what it is, we'll go ahead and grab one of those. These are the kinds of things that we try to prioritize.
That's
Chris, you
not
want to add
lasers.
anything to
Yeah,
that?
please.
That's right. And a lot of the samples that we collect are used by researchers to explore and understand deep sea systems. So if they're animals, we often extract their DNA and describe and identify them to figure out what they are, to see if they show relationship to other species. Genetics is used to ascertain whether they might be the same species in other oceans, to determine how far-reaching some of these animals might be. For the geological samples, they will often take chemical analyses and look at the physical structure of the rocks and try and determine what their origin might be, how old they might be, and how those might relate to the underlying structure of the areas that we're investigating. The Guyot we're on is part of a larger island chain near Hawaii, and we use a lot of these to... Oh, what's that?
There you go.
Ah,
I know.
lovely.
That's a star, I think.
Believe it or not, we're looking at a hermit
I think
crab,
we're
folks.
looking at the backside right now.
Backside.
Parapagrid,
We can
I'm
start
pretty
here,
sure.
and then if it doesn't swim, we can zoom around.
So as we get closer, you guys are going to see something
Probably if
really
anything, want to rotate
weird.
more
'cause
around
that's what
to your
these animals
starboard
are.
side.
Just there's no other word for it.
Rotate
Weird.
starboard. Do you want to do that
This is the
right
deep sea
now?
equivalent
Yeah.
of the thing with 2
Copy.
heads,
Otherwise, we're going to see
except
nothing
it's
but
got
an enemy with legs.
more than that.
Some of you are already going, what is he out of his freaking mind? That's not a hermit crab. Good grief.
Good grief, Chris. Is that a hermit crab?
But you wait, you just wait, and you're going to see something
Biggest
really
ornament
cool.
I've ever seen.
Oops, sorry, video. You guys scraped. Yeah, my toe scraped some up, but let's see what we got.
There he is. You see the crab sticking out. You can see his legs. But what on earth
You're
is on the
rotating
top
on
of
you.
him, Chris?
Gosh, Chris. Well, I don't know. Why don't you tell me?
Well, I think it's a zoantherian, so it's not an enemy.
Holy
Instead
smokes,
it's-
Batman. What's a zoantherian?
I don't know. It's a type of cnidarian.
Current's
It's
just
in the
gonna be
group
blowing
called
all this
Hexacoralia
into it. One on
because
the other
it's
side would be better.
Yeah, video. You want me to try
has
to
arrays of
look at his
6
face?
instead of
You can try.
Opposite I went, but
tentacles
Well, he
around his
rotated on you
mouth,
about
but
90
more than
degrees.
6, but it's
He's going to
just
walk
the-
on you now.
Yeah, video, but if nothing
The
else, we'll get
arrangement.
the
bridge move complete.
That's
dust
right.
cleared
So
up. If we move around to
this
the
is a
other
hermit
side.
crab. For those of you at home who've perhaps never seen one, this is similar to the little crabs that
I'm
you
gonna
can buy at
kill
your pet
lasers,
shop.
That's all right.
And the
Sure.
ones that you see in shallow waters and sometimes even on land, they carry a little snail shell around with them. These are different. You can see there's the claws and there's the legs, but these are different because they, instead of carrying a shell, they actually carry this kind of amalgam of sea anemone-like
Sit down and
animals
get a
called
super stable on in here
zoanthids.
centered.
And so what you're seeing there is
I'm gonna
sort
rotate
of
to
1, 2,
follow
3,
you.
4, 5 of these
Copy.
polyps of these zoanthids that form this kind of pseudo shell. I believe I've been informed by other people that this varies by different groups, the kind of replacement that happens. This is actually something that is seen not uncommonly in the deep sea and in other places where the
Ready
shell
for
is
tight?
sort
Yeah,
of
go for tight.
replaced or
Center him up.
Centering.
yeah, replaced by sea anemones or some other kind of cnidarian. And this is presumed to be some kind of relationship that is a
Sediment
win-win.
still coming in. I got the black super crush right now.
Sea anemones get transport out of it, and the crab gets some kind of defense, because each of those polyps has a number of feeding tentacles with stinging cells, so even some of the food that either one of these eats might be beneficial to the other. So definitely, looks like there might even be another polyp underneath it.
Copy that.
Yeah, and by the way, I call this a Parapagurus. It's a Parapagurid. It's in the family Parapaguridae, but I think it's best ID'd to the genus Sympagurus, and it looks quite similar to something we call Sympagurus burkenrodii that has been identified from Hawaii, so not that far from here. But I think going to genus here, Sympagurus species, is a pretty decent ID for him. So that's not weird, is it? No, not at all.
Off to my port
No, Chris, not weird.
I'll center with you, co-pilot.
Had some type of little sponges or something next to it.
Do you want to zoom on those watch leads?
Yeah, it's been a number of years since I've seen that hermit crab, so that's quite a treat.
Well, actually, do you want to zoom on those little sponges?
Which little sponges?
Oh, sorry, I thought I heard someone say little sponges.
Yeah, I think we can just go on.
Copy.
With that. Yeah, thank you. Oh, Sean, it looks like you made another mark there, pilot.
That one might have been there.
from the previous D2, right?
Yeah, I think so. Now I'm coming in.
It's a different little vase.
Sean was just making his mark there.
Sponge coming
Sean
center.
Kennison was
We
here.
snap on it.
Yeah, absolutely.
Another one of these Semperella glass sponges that was off to the left, and here we got something a little different. This is worth a zoom, if we could.
Oh, this
is a different type of glass sponge. Again, it's a sediment specialist, and this is going to be in the family Hyalinimatidae, and I'm pretty sure. Well, for the moment, I think we could
lasers back
probably go to the genus
lasers,
Hylanema, excuse me. So it does have this fibrous anchoring system, but in this particular case, they're kind of interwoven into a stalk, and then they actually grapple down into the sediment at the bottom there. And so slightly different way of attaching. This enables the sponge to get up off the bottom a bit more, since it's a smaller guy.
Okay, video's clear. Stripe it
filter
with
the
lasers.
water column for hopefully all types of nice, tasty tidbits.
Got lasers. Video's clear.
Great.
I turned to port, Sean, so
copy that
a little bit. So I'm gonna kind of work it back to north.
Yeah, copy that. Just leave me. Oh yeah, looks like something's crawling or something.
There's a dead sponge, two dead sponges.
Okay, I'm lined up on north now, Sean.
So they're in the process of becoming kind of that little nothing more than the roots that we saw earlier on.
So
serious
view
be white.
past that sponge to the left
this is a
shows
dead one that's brown, probably because of accumulation of sediment that it's not able to clear
dark circle
because it's dead.
Yeah.
And then eventually that tissue will deteriorate, and there's a dead stalk right there to the left of it. So it's just the process of how these animals decompose once they die. Pilot, yeah, there's a weird circle.
Yep, making my way. We'll find out what that is.
Huh.
Neither one of us is about to hazard a guess here, are we? I don't know what that is.
At first, it looked like it might have just been some illusion, but you're seeing it too.
I think I see something swinging around the
final
center.
remnants of a sponge base, and we're seeing sort of an arrangement of nodules that were somehow around.
There's
something. Oh, it's a black coral. Look at that.
Huh? That's weird. How it's
how did it do that with the nodules? It swept across them.
Could that be
Yeah.
Yeah, but how does it move nodules? Look at the size of this.
Well, I don't know if it's actually. I don't think it's that the nodules are different. It's sweeping like a brush.
It's clearing the nodules around.
It's cleared off. Yeah, I don't know if that's deliberate or if it's just a current thing, but it looks like all of the organic materials on the surface of these nodules has been brushed off, and so this. Like a feather duster, as Steven Oskovich is saying to us from shoreside, it's like a feather duster on the rocks. So, yeah, that's kind of weird and neat. I don't think I've ever seen that.
Base is slightly hidden by the stalk.
That's kind of an interesting adaptation if that's deliberate.
Let's go up to the stalk.
All right, Scott, Tina Molitsova is not with us right now. You're on the spot, buddy. I know you worked on black corals, and we need some help on this one. Come on, call in. We're calling a friend now. We're not going to use our 50-50
yeah, you're
yet.
fine. We're done moving, so.
So we have a pool of shoreside scientists who are among the world's experts on different types of corals and other animals. And so a lot of them are currently dialed in to a chat room that we're messaging with on a constant basis. And so they're able to view the video just like everybody else is, and they're able to offer their professional input to us almost immediately. And so they're watching these and able to give us their professional opinion. Some of these people, some of these scientists are calling in from Japan and others from other countries as well. And so we've just observed something that is really remarkable, this black coral, which we believe is somehow brushed into a sort of like a feather duster, brushed all of the sediment off of the nodules in a circle. And so maybe it's feeding. Perhaps it does that for other reasons, or maybe it's accidental.
The dust is on the north side.
Yeah, so Scott France has gone ahead and put in an ID in the event log. He apologizes. He can't call in right now based on where he is, but he believes it's possibly in the genus Schizopathes, which is a black coral species. And we've seen some of the members of this genus on sediment where they pure sediment without nodules, where they actually sweep a clear area as the currents rotate them around in a perfect little circle around their bases. So thank you very much, Scott. And I also see that Amy Bacco-Taylor has joined us from Florida State University.
Yeah.
So she's another octocoral specialist. Thank you for joining us, Amy. Yeah, so we've got... I guess
we have a tiny sea anemone in view right now. Is it a sea anemone?
Yeah, I think so. Yeah, I don't think it's a cup coral. I think it is an anemone.
All right. Come on out. Keep moving.
Lasers across the coral?
Yes.
That was lovely. Thank you.
You are welcome. Good for lasers?
Okay.
Great. Wow.
I'm lined up on north still. So.
All right, we're going north.
That could only have been improved if a starfish had come along and eaten it.
I bet the coral wouldn't say that.
Yeah, I know that. A lot of animals, Chris's favorite animals are evil beasts that like to eat them slowly over time.
Just like you and the chocolate sundaes, Chris. Oh, this is interesting. Is it a
Some
sponge?
of you kind of are wondering, what? Sea stars? They just sit there on rocks. What are you talking about? But down here in the deep sea, they can be pretty voracious predators, and even in shallow waters, I imagine.
Oh, yeah. Sea stars are important predators in many
crab in the upper
tropical
background
to cold water systems.
Just left of center.
Sometimes they feed on mussels. Sometimes they feed on corals.
The white
and down here, they feed on a bunch of things.
I think it may be another one of these piles, but it looks like another
Sako Matsumoto is calling in from the Chiba Institute in Japan. Also noted that we saw a similar one of these Schizopathes around the same depth last year, 2,600 meters.
Optic illusion, sponge base.
Copy that. Not a crab. Could probably write that down.
So I think pretty soon we're going to start seeing some rocks. Just looking over the monitor at the navigator's screen there. So we can see the contour lines kind of forming up a bit.
Yeah, the squiggles.
Sirius, yeah.
Squiggles. I haven't seen any subjects yet.
They're obvious from Sirius view. Yeah.
Kind of
I don't see anything.
Yeah, except I don't see. Sorry, I don't see Stream 3 in front of me right now. Where is it?
I could do it. So just for those of you out there watching on Stream 3, we have what we call the quad split. On the upper left is the ROV primary camera. On the upper right, you have the ROV pilot's graphical user interface, or the GUI.
Copy.
The bottom left of that quad split is what we call our high packer. That's our map of the ocean bottom in relationship to the ship, the ROV, and Sirius.
I think you called it now. We have a 5-minute delay.
And on the bottom right of that quad split is the ROV sonar split screen, which left side is the ROV, the right side is the Sirius sonar.
Yeah, thank you, Roland.
30 meters due north, decimal 2.
So that was the voice of Roland Bryant, who's on video today. He's on video for a lot of these dives. He's a master at
yeah, I see
getting you some amazing video.
Squiggles on the starboard, but
and more of these Semperella sponges.
We can modify that quad split as the shoreside science. You guys
right here.
They're easy to see from my view, but
yeah, they're not as obvious from D2's view.
Yeah, because looking down and I have contrast.
I mean, that looks pretty.
Oh, this is interesting. Look, there's a trail in the
try to find the end of the trail, maybe.
Bed.
I think it's immediately right of lasers, but I don't see any creatures next to it.
Some animal moved through there and displaced some of those nodules, clearing them away as it slowly meandered
it might
about.
even still be in there somewhere.
Probably one of your sea stars ate it, so it's no longer around.
Watch Aid's interest on Zoom on this?
No, thank you. We've
Copy
seen this one already. Thank you. This is another Semperella, which you got some good images on earlier.
Star at the bottom of it, pink.
Star at bottom.
Let's take a snap on the pink at the base of it.
All right, quick snap on base.
Yeah, and actually you got a couple things here on the base. What do you think that orange thing is, Chris?
So this orange animal is an animal distantly related to sea stars. This is a brittle star, probably in the family Okeanos. I can't see the disk, but it's probably something like Ophioplinthaka. These are commonly encountered in the deep sea, and I'll annotate.
Thank you. There's no way we're going to get that one.
It's probably residing amongst all of those fibers as defense, but they're often seen on sponges like this in great densities. But their relationship has never really been.
Video's clear.
I'm not aware of exactly what the relationship
I think they're happy. Move on.
So these kind of nodule beds that we're traversing across before we actually get to the steeper part of the ridge, these can be common in certain areas of the abyssal seafloor. And as I mentioned before, there's a group of scientists, Dr. Craig Smith at the University of Hawaii and Diva Amon, who's currently, I'm trying to remember where she's wound up working now. She just moved recently. But they specialize in these communities. And they actually put out a recent paper on nodule communities, I'd say about a month ago.
That's right, and these types of habitats, these settings are found all over the world, and sometimes you see sea cucumbers on them and various types of filter-feeding sea stars. Many different kinds of animals, fairly large
more often
as
in
well.
The distance.
And so here, but
the
occurrence of these types of animals is variable across the world. So here we're seeing sponges, but this could easily change into sea cucumber land or some other type of sort of species-rich area before too long. But again, just because we're not seeing it doesn't mean that there isn't something here. And if we had if we were physically able to transport down to the surface and pick up a sample and look through the nodules and all of the sediment, there's conceivably a huge diversity of worms and other microanimals, or even protists that are living in the sediment and amongst all of those nodules.
Rail cam's out. I'm not sure. Co-pilot?
I could cycle it.
Maybe it's looking right into this. No. It's not looking into Sirius. I don't know.
ROV nav, watch lead 1.
I'm going to cycle power to starboard.
Seeing any rocks out there? I kind of looked over your shoulder and it still looks kind of flat. Is that right, or am I missing it?
Yeah, watch leads. We've been keeping the ship move steady to try to get through this.
Yeah, I mean, this is interesting, but some rocks would be nice. Great, thank you very much. Yeah, you have been. I've noticed. Thank you.
So one of the things that we're often looking for on settings like this are rocks and other large structures, because these often form habitats for animals in the deep sea.
Taking into account
These are
other previous move
animals that are extremely opportunistic, and so there's a lot of harsh environment to deal with in this setting.
Have 5 minutes
and so we're often looking for places where these animals are either finding habitat or perhaps
up to what's that, 40
finding
meters?
Potential food items.
But we might move another 15. Plus, if we do another 30, that's already 45.
So Scott France suggested that we kind of mention to the public here about how cold it is. It is 1.69 degrees Celsius, and I can feel it in the cold room, in the control room right now. It is cold. Oh, wait, that's the AC. I'm sorry.
40 meters.
The actual water temperature down there is 1.69 degrees Celsius. I don't know what that is in Fahrenheit. So you specialize in math, Chris. Come on, do the conversion. Oh, you're looking at me.
I specialize in math.
0 meters, bearing 000.
It's cold.
It's cold.
It'll hurt your hand if you put your hand in it.
Which we did yesterday to get the sponge out of the collection box. It is cold.
I've put my hand into freezing water, both collected from the deeps and when I've been in Antarctica. And let me tell you, nothing's quite like putting your hand into just freezing water. It's not just a little uncomfortable. It's very, very, it's almost painful.
No, it is painful.
Yeah. It is after a long period. That's for sure.
Bridge move of 40 meters, bearing 000, speed decimal 2 initiated.
Copy.
Of it is there's extreme pressure down here, probably greater than the inside of a fully charged thousands of pounds per square inch. Thousands of pounds per square inch.
So we're currently at a depth of about 2,400 meters.
We're at just under 2,600 meters.
Right. Most people don't have a sense of just how deep we are. At 1,000 meters. That's obviously less than where we are now, but 1,000 meters is about the height that kind of depth is 5.5 Washington monuments, which I calculated before I left Washington D.C. So at this rate, the depth of 2,400 meters would be at least 11, 12, 13 Washington monuments from the surface. That's a lot of Washington monuments stacked tip to tip from the bottom of the ocean to the surface of the ocean. So we're in deep, and so it's very cold, it's very deep, and it's very dark. The light you're seeing projected from the ROV is something that is artificial. In normal circumstances, if we weren't here, everything that we would be seeing would be pitch black. And so this habitat is a very sort of cold and unforeboding one, and so the animals which live here have very unusual specializations or are very adapted
quick,
to this environment.
Zoom? Is this anything?
Video zooming in.
Oh no, that's just overturned. All right, never mind. Back out.
Like a cleaned off.
Yeah.
Nodule.
All right, looks like we got some tether.
Sean was trying to compete there. Our pilot Sean was trying to compete with Roland Bryan, who has a reputation for having the sharpest eye for things on the seafloor. But
it's quite the opposite for me, apparently.
Well, basically, Roland beats just about everybody on the ship. He's got amazing eyes. One thing I wanted to point out, since we've put out some information about this, is the oxygen levels that we have seen today as the ROV was descending through the water column, because Johnston Atoll area here essentially has a shallower oxygen minimum than the Hawaiian Islands, particularly the Northwest Hawaiian Islands in the monument up there. And so I'm looking at the CTD profile from this morning, and it looks like the O2 minimum was hit at around 600 meters. So it's a little bit shallower than it was yesterday. It was about 650 meters yesterday. We progressed a little further south.
Still nothing on
and we'll continue to go south, and I expect that oxygen minimum to continue to climb. And it's probably going to get...
Do the contour lines get closer together?
Shallower than 500 meters, might even get to 400 meters at some of our more southerly seamounts on this cruise. So what am I talking about to the general public? Well, you know, light, as you go deeper, it just gradually, or rather precipitously, extinguishes, and then it just stays dark. Okay, it just doesn't get any brighter as you go down. And temperature's kind of the same thing. Temperature kind of drops off the deeper you get, and then it finally levels out just above freezing in most places. But oxygen does something weird down here. So it can actually take a little hike up, and then it starts dropping down, and then it reaches what's called a minima, if you will, and that's the lowest point of oxygen at a particular site, and it's usually can be anywhere between, on the continental shelves, it can get up around 200 meters or so is where you see the minimum, and up in the Northwest Hawaiian Islands, you see the minimum at 1,000 meters. And by minimum, I mean that once you go deeper than that, it starts going back up again. So it's a really cool thing. We pay attention to it because we think it's possibly affecting the development of communities down on these sites. So a really interesting phenomenon.
That's right. All animals need to breathe, and so oxygen remains an important limiting factor for the different kinds of organisms that we see, even at depth. And so these are the things that deep sea biologists often look for, is patterns that affect the distributions, how these animals occur in these different areas. And we don't get to see them sort of firsthand and in the big picture. So we often look for sort of chemical or environmental factors that correlate with distributions in the hopes that that will show a correlation.
You got a fish?
Yeah, I think we might have a fish.
Yeah, I see it.
Yeah, thank you.
Fish.
Or something moving.
Yeah.
Yeah, I bet it's a...
All right, video, as I get closer, feel free to come in.
What have we got here? It is a fish. Oh, I think I know what you are. And Bruce Mundy is not here. Ken Sulak, are you there? No, I don't think so. But I think we're looking at something that is possibly a duckbill eel or a sorceress eel. I think it's a sorceress eel of some kind.
A what?
A sorceress eel. And this is a bit different than the ones we've seen elsewhere. And this is quite a sighting. These things are rare. And so this is a real treat. And I've got to actually go up and look at the specific scientific name on this thing.
Keep it on my tether.
So it seems like we've lucked out and found an extremely rare sorceress eel. As a starfish expert, I don't know what that is exactly, but I'm sure Chris Kelly is about to tell us.
I am about to tell you if Bruce Mundy, who's our resident fish expert on most of these cruises, unfortunately Bruce is at a conference right now.
Ironically, the ichthyological and herpetological conference.
Right. But anyway, this is in the family Nettostomatidae, which are the duck-billed eels family.
Yeah.
And this type of eel is probably in the genus Vinifica. And if I was Bruce on Knot, I could tell you why it's named the sorceress eel. It has something to do with its schnozola. There's some weird ornamentation, I think, going on the nose, which you can sort of see.
Yeah, sorry, video. He's
giving us a look.
It's fluid.
This is something else. This is great. A fantastic find here.
Wow.
That's probably the third one of these eels we've seen in the 3 years that we've been doing Capstone out here. So
wow, I consider myself fortunate. Everyone watching can also consider themselves fortunate because this is, as Chris just said, the third time we've probably seen this species during this Capstone expedition, which began in 2015.
I'm going to let him swim away, video.
Thank you, pilot.
He's going in black. Great.
Oh, that's a beautiful shot.
Down.
Yeah, so Kelly Elliott, for those of you in our event log, has actually put in a link where
I'll try to line back up on northbound.
And I think this was
center up.
In 2015 when we saw our first one. He actually did an update or a blog or something. I've forgotten what it was, the format, on this particular eel when he describes it. And I'm going to click on our link right now and see if I can find it.
And so this is a lot of what deep sea exploration is like. Sometimes you have, it's like waiting for the bus. Sometimes there's nothing for a while, and then suddenly you find really interesting things, or 3 of 4 things at a single moment. So you never know what you're going to encounter. So it goes from 0 to 60 very quickly.
Copy. How's move?
Looks like ship is stopped.
Do you know when it ended?
Looks like, judging by USBL, got not too much left to swing. I think we could call another one in, John.
Great. Yeah, put in. I think 50 is fine.
Okay. Keep you out front then.
Go ahead, RV Nav.
Ready for a ship.
Roger.
50 meters, 0.029.
That is 50 meters, bearing 000, speed decimal 2 knots.
Good copy.
So if you've just tuned in, I'm Chris Ma from the Invertebrate Zoology Department at the Smithsonian's National Museum of Natural History, and I'm here with my colleague and co-biology lead, Chris Kelly from the Hawaiian Undersea Research Laboratory at the University of Hawaii, and we're part of the 2017 NOAA Laulima Okamoana expedition to the Johnston Atoll region. And we're currently at a depth of around 90 meters, and we're surveying a guyot that is currently unnamed and looking at an unexplored bottom surface. We just saw a very rare, rarely seen anyway, fish called a sorceress eel. We're currently traversing a field of manganese nodules, and what you're seeing off in the background are several glass sponges that are apparently the dominant large animal in this area. These are sponges that are made out of glass. They're probably also homes for other smaller animals such as brittle stars. But a lot of the things that we're seeing in this kind of setting are probably very small and so not immediately obvious. We've seen several tracks in the nodules on the bottom, suggesting that there might be snails or other kinds of animals residing amongst the texture that's on the bottom there. And so who knows what we'll see next. Stay tuned as we proceed along the bottom. We'll be sure to comment immediately as soon as there's a new update.
So we've moved beyond that particular eel, but I advise those of you who want to get some more information on this to go to the mission log from September 1st, 2015 on the Okeanos Expeditions website. And Bruce Mundy has got a great little mission log that he posted. It's called The Sorceress and the Scientist.
Shows them better
And you can get some other information on it. And he actually went and researched this and determined how many species and so forth that are on here. Really nice piece here. So take a look if you want to find out some more about that really incredible eel. Okay, some more of these glass sponges in the genus Semperella. This is clearly the dominant sort of megafauna, if you will, that's on this nodule bed. Haven't seen a whole lot of other things, a few small items. We had that one black coral, that was pretty cool, and of course the eel, but
and I've actually taken the liberty of posting the link to that sorceress eel from our recent observation on my Twitter at Echinoblog, so you can go and check that out on the hashtag Okeanos on Twitter. For those of you who are following us and taking screen grabs.
so I don't know, Nav, you're taking a while to find us some rocks there. I don't know what's going on up there.
What distance has the ship moved since we landed? Meters? What is that? 150. I'd like to keep a couple minutes between moves.
Can you move the map down so I can see what's ahead of us? Thanks. Okay. Gotcha.
So, Chris, I'm noticing that all of these sponges are present on sort of a sandy patch. Are these all rocks that have just sort of risen above the nodule field and are covered with sediment, or are these just little patches of sediment that have built up above the nodule surface?
Yeah, I think this is the roots have accumulated more sediment and have created a mound. Even these areas where the dead roots are here, I think that's what you're sort of seeing.
Right.
The roots actually create kind of like a teepee effect, and then the sediment fills in whether the sponge is there or not.
Oh, so you think this is created by the sponges?
Yeah, the sponges. It just, it's.
So that's interesting because the sponges then create habitat for themselves in a way. Oh yeah, let's take.
If we could look at. Well, there was a different sponge there to the lower right.
Yeah, copy that. Going back.
Sorry, I didn't suggest it because I thought it was one of these Semperella leaning over, but now I can see it is different.
Good morning. It looks like our wind shifted
know what
a
this
tiny
guy
bit.
is.
I would like to adjust our heading about 10 degrees to starboard to a 070.
And that is fine with us.
Roger.
All right, video. Go for zoom.
Video zooming in.
So we're zooming on a glass sponge.
I don't think so.
Yes, we are.
All right.
And this is going to be a new species of in the genus Sericalophus, and we're calling it right now Sericalophus new species 1. Dr. Henry Ricewig in Vancouver, one of the foremost experts on glass sponges, collected one of these off of Hawaii back in, when were you on? It was
1999, 2000,
yeah.
2001.
Yeah, it was a cruise that you were on the Hurl submersibles.
Oh, wow.
And yeah, this is really weird, and it's called a catcher's mitt sponge, but this particular species has these spicules that are coming out of the face of it, which is really weird, unlike the shallower water version of the Sericolophus in Hawaii that's called Sericolophus hawaiiensis.
So this is a glass sponge, and one of the things that characterizes this group is that their skeletons are actually made of glass, silicon dioxide. And so when we were panning across the bottom surface, you'll notice that all of these fibers, what Chris called spicules, these are essentially skeletal elements of the animal that they're basically like long glass fibers, like fiberglass or like fiber optic cable, and that's presumably how this animal is anchored to the bottom.
Thank you very much.
To the bottom of the sea here.
Watchers, are you happy with this?
Yes, we are. Thank you.
Great. All right,
much appreciated. Thank you.
Out as
and I now see that the cavalry is arriving. More cavalry is arriving. Tina Molitsova from the Shirshov Institute in Moscow has now joined us. So great, Tina. I'm sorry to say that you missed a really cool Antipitherian, but you're going to have to look at the images afterward. And Scott helped us out. He thought it was one of these possible schizopathies that was actually cleaning the nodules in sort of a ring as it swept around. I guess that's the way to describe it.
I thought that was really amazing.
Yeah. Very, very cool, but hopefully we can see some more black corals. Tina is an expert in a variety of different groups, black corals being one of her specialties. So she also does tube anemones, expert in that, anthomastus and mushroom corals, and a bunch of things. So it's great to have her in the event log.
Is it just me, or is there like a track through?
Yeah, I see it
nodules?
too. I don't see anything that seems to have made it though.
Okay. It's just you, Chris.
So as we've observed before, again, just because we don't necessarily see anything big and moving around doesn't mean that there isn't something there. And that there isn't something there. And so these tracks are probably left by some snail or some other kind of animal that is present among all of the nodules. And it's left a nice trail behind it, but something that it may have buried itself and made itself essentially invisible to us. It's hard to say, but or it could be made by something else, like a fish, just before it's taken off and left the bottom. So these are the kinds of interesting things that we see on the deep sea. Sometimes traces of animals, as much as the animals themselves. These are the types. There's a whole field of paleontology dedicated to studying things like this, trails, ichnos, and the study of ichnology.
Center up. Tell me if it comes back.
I think there's a German word for these, Lebensspuren, the living tracks of different kinds of organisms. There's actually a labeling system as well that is parallel to living species. They actually have a code of nomenclature which names these in a very similar fashion to those of living
I'd be
animals.
okay with another move, Sean.
Yeah, you're happy?
Yeah.
All right. How far else it void? 40 meters. All right. Yeah, you would do 40 meters. We can go to the void. I think 40 is fine.
Go ahead.
Hey, we're ready for another ship move.
All right.
40 meters, 000, 0.2 knots.
That's 40 meters, 000, speed decimal 2 knots.
Good copy.
Oh, what's that? Little.
Oh, the red shrimp.
It was a shrimp. Ah, okay.
Just off to my starboard pilot.
Bearing 000, speed decimal 2 initiated.
I think this is potentially another
Do a quick zoom on shrimp video. I don't want to go too far over that way.
Certainly looks like it. So there's some types of shrimp, and you'll probably see this as we continue this cruise, where their legs, they tend to swim with their legs out lateral from their body. Kent the Fire is one of these shrimps that actually paddles more right underneath its body, so you can tell it's different on that basis. But also, I sort of refer to it as a drunken sailor shrimp, because some of the shrimp, you see them moving, they're just kind of powering along in a nice even path. And this guy is just upside down and curly-cueing and do all types of weird things. Like he doesn't know where he's going. And so it's kind of sometimes the behavior of the animal can help you to identify what it is.
All right, come on out, video.
So if you've just tuned in, this is Chris Maw from the Smithsonian's National Museum of Natural History. And next to me is Dr. Chris Kelly from the Hawaiian Undersea Research Laboratory at the University of Hawaii. And we're part of the 2017 NOAA Okeanos Explorer Expedition Laulima Okamawana. And we're surveying Johnston Atoll
you could probably just
in the Central Pacific. We're currently at a depth of about 2588 meters below the surface, and we're looking at a deep sea nodule field currently occupied by several glass sponges, as well as several other interesting forms. We just saw a while ago a rare sorceress eel, which hasn't been seen very frequently. This is the third sighting of this eel since
what time is it?
The Capstone expedition began in 2015. And there are a number of other animals smaller animals that we've seen amongst the sponges and on the sediment.
I'll just set real quick.
So stick with us if you're getting tired of nodules. I'm looking over at my monitor, and I can see the contour lines are constricting just up ahead. We also saw what looked like our first rock, a very small one on there as well. So I think very shortly we're going to start getting into steeper terrain.
So as Chris said, get ready to rock.
Oh, jeez. Doing a little bit of a seat change here. I guess that everyone's given up on Andy finding the rocks for us, so they're going to put in a new navigator here.
No, I'm going to find the rock and pick it up now.
Yeah, there you go.
That's a good point. Actually, we are also here to study geology, and so at a certain point in our future expeditionary study, we will collect a rock. Many rocks are used through chemical analysis and through physical analysis by looking at the structure of the various clasts in different rocks to determine the history of the surrounding geology. And so our study here is not just to look at the animals, but to investigate the geological history of a lot of the underlying substrate and the island chain and the earth, the geological structures that make up this region. So characterization of these new areas is a priority of our expedition.
So Tina Molodtsova has apparently seen a picture of that black coral that we saw earlier that we thought could be a possible Schizopathes, and she actually thinks it's something that I have never heard of before. She thought it was an Alternatopathes, and Scott hasn't, I don't think he's heard of that as well, so this is something new. We're seeing if Tina can, let's see, her latest comment in the event log, yes, we just invited, it used to be part of Umbellapathes, so very cool. Thank you, Tina. And thank you so much for correcting Scott. He gets me all the time. It's great to see someone get him back one of these days too. So that's terrific.
Is there maybe something out there? Sonar? It's a little divot.
Yeah, maybe.
See in Sirius's view a little bit change in sediment looks of it, but you're just off to my port, pilot.
Copy that.
So we're getting a little bit of a change of terrain here. Oh yeah, if you folks look at stream 2, you can definitely see the transition we're hitting right now.
A little more scattered.
So we are getting some kind of more consolidated substrate here. It's relatively flat. I wonder if we could zoom in on that whip coral that's about to go off to the left.
Absolutely.
Great, thank you very much.
Looks like ship is still moving. Okay.
Yeah, ship is I think 5 meters left.
Okay, excellent.
Yeah, 5 meters.
So there's actually another coral after this one too that's to the left as soon as we get through this one, and this looks like it's going to be a bamboo coral.
Oh, neat.
ROV nav bridge, move complete.
So unfortunately Scott isn't on the phone. That's why I can poke fun at him, because he can't defend himself very easily. But we're going to be monitoring his comments and Les Watling. Both of them work on the bamboo coral group right now, and see if they've got a possible ID on this thing.
That's Maxim.
So bamboo corals are a type of octocoral. You'll notice that if you look at each one of those feeding polyps, there are 8 feeding arms around the mouth. And these are part of this colonial. This is a colonial animal, and so all of these mouths feed and contribute to the whole. And you'll notice that the reason that they're called bamboo corals is because there's a primary stalk, which is sort of a porcelain-like hard part that forms the structure that is the main part of the animal, and there are black internodes between a lot of the sort of the white elongate structures. And so when these animals are cleared of their tissue, that gives them a kind of a bamboo-like appearance. These are the skeletons of these animals. Is actually an interesting thing that most people don't realize, is that the skeletons of these are almost like porcelain. And so even though this is this seems like, and so these are very fragile animals, although probably a little more resistant than one thinks. But you can see the skeleton is translucent, and you can see the skeleton through the tissue that is present along the stalk.
A little bit of time. I got you at
all of
70
these very
degrees.
Hungry mouths projecting into the water, trying to find
out
food.
The swing. So.
Again, 8 tentacles around the mouth. There are little sort of pinnate structures, sort of little fine hair-like structures that are coming out of each of those 8 arms around the mouth.
Okay, a little comment reading to you some of the information that's coming in through our event log. So Scott and Les have been discussing this one, and they believe it's going to, once this new genus is fully described and published, they believe it's going to be in a genus called Taeniostea, or Taeniostea. I'm not sure how to pronounce this. So
that's just as well. It's
so
not.
It does not exist right now, so that's not a valid genus. We would normally, at this point, probably call it something like Lepidopsis, an older genus, but because of the very long spines between the tentacles. But it will be moved on the basis of genetic information that Scott and Les have been acquiring. Abby Laponte, who's a grad student right now for Les Watling, is working on these corals as well. So there's going to be some major revisions for this whole family in the near future.
Launch leads, are we good on this one?
We're good on this, thank you.
Okay, come on after you.
So great. This is why it is a cooperative effort here. We needed a lot of help on that one, so we really appreciate people giving us their input, whether via audio or in the event log. So thank you very much to you two for the identification. I think we got a coral right on, I think you see it already because it looks like you're homing right in on it. Right on the top of this little boulder feature.
Dead ahead.
It's like a truffula tree. Maybe a possible chrysogorgia.
Oh, this is nice.
I'm pretty sure I know what it is, but we're going to get a little bit closer.
And I believe there's also a shrimp on the on the side of the rock closest to us.
Let's see if we've got some help already.
Come on in, video.
Ah, pretty.
Yes, yes. So Scott France has already commented that the pilots are already the pilots had already identified this, whispering this as a chrysogorgid octocoral.
Next question.
That's what it is. I don't know if we can go further than that, whether it's chrysogorgia. I do not think it's metallogorgia, but I'm waiting for Scott or Les in particular to comment, since this is another one of their specialty groups. They are zooming right now at a squat lobster. These are commonly associated with chrysogorgids, and this lobster is probably in the genus Europtychus.
And so I add further detail. This is again a coral called chrysogorgia. It's an animal, not a plant. And if you'll look closely for those looking in, there are 8 feeding tentacles around each of these little feeding polyps on the body. And so these polyps are much smaller than the one that we saw on that bamboo coral just a moment ago. And also you'll notice that the stalk differs in composition. It actually appears almost shiny, kind of golden, whereas the other one had a prominent sort of tissue-like covering over sort of a black and white porcelain-like stalk. And so it's characteristics like these which scientists use to distinguish between all of the different types of corals, and these are a subgroup of corals called octocorals that we observe here in the deep sea.
Is Watch Lead happy with this?
Yes, we are. Thank you.
Great.
Scott France from Shoreside reminds us that chrysogorgia translates from the Latin as golden coral, and that's likely owing to the very shiny color that comes off of the stalk. I'm sure there's a better word for it when one looks at them close up.
So we've got a little bit tighter ID on that chrysogorgia that we just came off of, and Les and Scott are suggesting that this could be something close to, if not chrysogorgia averta. So thank you very much for that ID. We're going to go ahead and put that in our C-Scribe annotation logs.
If any of those rocks are loose.
Some of these might be.
Some lasers.
Sure.
What is that brown tube?
Well, there's a coral right there too. What brown? Oh, that's a dead sponge stock.
Oh, okay.
Yeah, maybe. But there's a sort of a wispy coral behind it as well.
Near or kind of?
Near. It's almost center right now. If you keep going forward, you'll see it.
Up, maybe up here.
Yeah, it's about center right now.
Oh.
Right above the lasers.
Okay, yeah, that's fine.
And there's a nice coral in the back there.
Cool geology here. It's like a...
Yeah, so we're starting to get into some animals now as we get into a little more consolidated terrain here, which is a pretty common observation that we make. A lot of these larger megafauna tend to need sort of a little more durable substrate to attach to so they don't fall off or fall over or anything, so you don't see them very often in the nodule beds. And then once you get onto this hard rocky surface, then you start to see things. So I've got an idea on this one as well from here, and I'm going to, of course, defer and wait for the experts out there. So Scott and Les, I concur, they're the ones who really know, but I concur with them that this is another type of chrysogorgid coral, but in a different genus. It's in the genus Pleurogorgia, and it looks very similar to the species Militaris, but Pleurogorgia species is fine right now. We've collected a number of specimens of these over the last couple of years. And we've seen them not only in the Hawaii and Johnston area, but also, if I'm not mistaken, down in American Samoa. I believe Santiago Herrera collected a couple of these down in American Samoa as well. That's at least in this genus, maybe not the same species.
Okay, you can come back in video.
So they seem to be fairly widespread.
Oh, what's that thing that's further up on the branches? Ah, there we go. So it looks like on this, what was it?
Pleurogorgia.
Pleurogorgia. Yeah. We have a barnacle. Most people are familiar with barnacles as sort of shallow water fouling organisms, things that occur on the sides of rocks or on the sides of your boat. But they occur at great depths as well. And so this, I think, looks like some kind of stalked barnacle. There are two types, the ones that are sort of flat and they kind of look like acorns, and then there are another type which have little sort of fleshy stalks, and these fleshy stalks come up to these sort of shelled or protected areas, and then from those the legs of these animals are projected out into the water, and they use these legs to feed. So although they appear like mollusks, they're actually crustaceans. They're actually more closely related to things like crabs and lobsters, and so they have jointed legs which they present out into the water, and they use them to feed.
So Amy Bacco-Taylor has also concurred. So we now got 4 people who all agree that this is in the genus Pleurogorgia, so that's great. Thank you very much, Amy.
Watch these, are we good here?
Yes, we are. Thank you.
Come on out video whenever you're good.
Can we just get a full specimen?
Oh. Sure.
There was some interest in this bamboo coral to the right there. I wonder if you could do a quick zoom on that before you depart. Sure. I guess we could do this, given that we've got a couple bamboo experts on the line. What do you think, Chris? Yeah, I guess
we can take a look at it. It looks similar to the one we saw before, though. So
I don't know about that. It may or may not surprise you.
Thank you.
These things, there's a lot of these unbranched ones out there. This is a very complicated family. Looks a little different to me based on the density of the polyps. This doesn't look like it has nearly as many polyps coming off of it than the previous one.
Okay, let's go ahead, video.
And you might be rocking, right? I might take it. Well, I mean, could have been different. Let's see what.
So another bamboo coral could have been the same one before, but again, we're looking at a very interesting translucent kind of tissue. We're looking at these very cool 8 feeding arms around the mouth with fine hairs around them, and an interesting sort of orange pigment around the mouth as well. Very fascinating.
Yeah, pilot, that's good. I'm a little embarrassed right now, and so yeah, we can move on.
It's fine.
Yeah, so let's see if they think, yeah, it could be different, but they think it's the same. Okay. See, they said it could have been different, so I wasn't that far off.
That's fine.
Okay.
I mean,
and that they being Scott and Scott France and Les Watling.
Oh, that's really interesting. Oh, there's a nice
different type of Chrysogorgia there.
Any interest?
Maybe a snap zoom on this. Looks like it could be Chrysogorgia geniculata, I think, as one possibility.
That's max zoom.
Come on out.
Oh, wow. So is this...
Did you want a stationary zoom? Watch these.
Were we done with the Chrysogorgia?
Yeah, I think we're okay with the Chrysogorgia. Okay. Thank you.
All right, pilots, do you want any moves for preemptive Sirius motion?
In just a minute, I was going to have them scope out.
Push out a little bit first.
Copy.
Interesting terrain right here.
So Nicola has departed to go get some sustenance. It's lunchtime here, and we've got Matt Dornbach now sitting in her chair. And Matt is serving, among other roles, as the data manager on the cruise and is helping out tremendously with specimens and the specimen database and imagery that we're taking and so forth. So great to see you, Matt. Chris Ma has also departed to go eat. So this is probably when we're going to come across the Sea Star. And let's see if the prediction comes true. There is a stocked sponge there almost straight ahead in the upper
left. We go for another move, 30 meters.
Seen so far today. I wonder if we could
north.
go ahead and document that. Looks like another Chrysogorgia.
Slightly off to my port pilot, but
geniculata.
Got it.
Possibly. Let's see if anyone else went to geniculata.
I'll actually come look at it from over here. Just keep an eye on my swing arms if you guys would.
Yeah. Will do. I got your lowers kind of towed up nicely, so
ready for ship now? I am. Are you?
You have pretty good clearance.
All right, 30 meters due north at 0.2 knots. That's 30 meters bearing 000 at 0.2.
Yeah, Amy's having a few technical issues. Battery is running low here.
Watch these.
But
Do you want to
Scott
zoom here?
thinks it could have been a geniculata, but definitely Chrysogorgia.
Possible.
Now we're going to zoom in on this sponge, and yes, it's something we haven't seen so far today.
Okay.
So it's really great. We're going to get a close-up of it. So this is a stocked glass sponge, and in this case, it does not have the fibrous attachment to the rocks. Instead, it glues itself to the rocks. And this is in the family Euplectellidae. And there's a subfamily which has all the stocked euplectellids, and that's a subfamily Bolosomini.
Come on,
and that's what we're looking at. And I think Bolo is kind of in reference to kind of the round head on it. These are really beautiful sponges. They tend to have a lemony tint to them. There's an osculum right on the top, or sort of a cavity, if you can see sort of on the top of the left there, and that's where water comes out of the sponge. and water's entering the sponge from the sides. So that's great. We can record the presence of this sponge at this location.
Okay. No further zooms needed. Watch leads.
I'll go ahead and throw in a duplicate here. We can get rid of later on.
Watch it. Did I hear that we're good on this?
Yes, we are good. Thank you.
So video whenever you're good.
Gonna get a specimen view.
Something big off to the left, but I think it's just another one of these larger stock sponges, the Bolosoma. I think it's in the genus Bolosoma.
Okay, full wide.
Great. That might be slightly out of our range, but
I can turn to port.
Sorry, pilot, could you repeat?
I said it might be slightly out of range, but we can go take a quick look.
Yeah, even the distant ones, but I think that's what it is. It's just the grandfather of the one we just imaged. Yeah, I can already tell from here that's what it is, just a larger colony of the same sponge, Bolosoma. These can get very, very large. The first time I saw this particular sponge back in 2003, there was sort of a field of these, and this is up in the northwest Hawaiian Islands in the Papahanaumokuakea Marine National Monument. And it looked like there were giant yellow lollipops all over the seafloor. I was questioning what planet I was on at that time. It was really remarkable. So this is beautiful. We're getting a little better view of the head here, and see that big depression. It's called an osculum. And again, a lot of the glass sponges don't have color. This is one of the few species that does have color. It's really, really beautiful.
Whenever you're happy, video.
Video going full wide?
Yes, let's go. Let's come out.
Be advised, pilot, I turned to port slightly, so
copy that. I will follow you back.
We're going to have to come back to starboard.
So terrific. Thank you very much, pilot, for that. And yeah, we're definitely getting things, the number of animals picking up, as everybody can see. There is a crinoid. Maybe we could take a quick look at this thing.
Yeah, you got time.
Absolutely can. Just set up here.
I'm lined up on northbound.
Perfect.
So,
where are we in that move?
We're about 2/3 the way through.
Perfect.
It's been 4 minutes since I called it in.
Come on in, video.
Okay, well, we thought we knew what feather star this was last year. We were calling it in the genus Cerametra, at least the purple variants of this. This is a crinoid. This particular one does not have a stalk, and instead it grabs onto other types of animals to get off the bottom a bit, and you can see by the cirri, its feet, that it's wrapped around this particular bamboo coral. And this particular crinoid, I think, is in the family Xenometridae. But of course, Chris Ma is eating lunch, and he's the echinoderm expert. And of course, we're going to come across one of these while he's gone. But some of the clues and identification of these animals are looking at the feet. Some of them kind of use them just to prop, and others have prehensile cirri. That means it can wrap around things, which is what this one is doing, and that helps to identify what species it is. Also, the number of arms, whether the arm tips are naked or they've got sort of sub-branching to them.
I turned slightly to keep you in view, so once you're done with this, I'd like you to
But very good.
center up again
Thank
as
you.
we
We're
move.
fine back here when you guys are ready.
Sounds good to me. Okay, video whenever you're happy here. Let's go full wide.
So the current identification for this, an update on what we thought it was provided by Dr. Chuck Messing. And he's at Nova University, and he thinks it could be Sathirometra.
Do you like that?
Possibly.
Yeah. Okay.
So it's going to be a harder one to pronounce than Cerametra, but
thanks,
we're going to get used to it.
Thank you, pilot.
RV nav, bridge
Come on,
move
wide.
complete.
Coming to starboard.
Copy.
Rather smooth right here.
Yeah. Okay, my set point is due north.
Oh, look at all these sort of boulders we're coming up to now. Again, another kind of transition in the substrate, and we clearly are getting into some higher terrain. Now, it's not clear whether these are boulders or they could be pillow lava formations, and they actually look like pillows. They are. So we are actually seeing pillow lavas, I'm pretty sure. So, pilot, a lot of this looks really glued, but I think we're finally into some basalt, and as we move up slope, if you see a rock that's possibly collectible,
okay,
that would be great.
I will keep my eyes out. Like you said, this looks rather cemented here, but we will find something.
Yeah, great. Well, I think we'll probably see this for the rest of the dive, so just if you see something that looks possible. So some of you might think it doesn't, you know, these just look like boulders or cobbles to you, but they're covered with this crust material, and believe me, they are glued into the seafloor.
Yeah.
Would love to.
You can, even a vehicle that weighs 10 tons like this ROV does cannot wrench one of these things free from the seafloor.
Sonar's into 30 meters.
Yeah, that is clearly a pillow lava formation that's coming on screen right now in the upper right.
In front of you.
Those of you who have a slight delay, you'll see it in a moment.
Would you go ahead and, actually I can do it.
And pillow lava is how lava erupts underwater in the deep sea, and then when it comes out, it forms these toothpaste tube or toothpaste-like trails or blobs that cool very quickly because it's very hot and it comes immediately into contact to near freezing water.
Less.
How far out are you from it?
And can cool very quickly.
I'm okay with 20 meters.
Yeah, about 18 meters probably. So 20 meters would be fine.
20 meters?
Yeah.
Do you want to keep going north, or do you want
to? Slight to starboard. I mean,
we'll do 10.
I would say maybe 20.
10, 20, 20?
That's what I'm looking at. Right
20
here.
at 20 meters? Is that? All right, put it in.
I can line up with this return here.
Okay.
That's right about 15, I think. 20, somewhere in there.
Yeah, let's keep it 20.
Sweet.
Go ahead, Nav.
Hey, you ready for a ship move?
I am.
20 meters at 020 degrees at 0.2 knots.
So that's 20 meters at 020 at 0.2 knots.
Good copy.
Couple more of these Bolosoma stock
I'm lined up on
sponges.
020 pilot.
That's a copy.
Again, with that distinctive yellow stock on them, which allows you to actually identify them from quite a distance because they're just, there's really nothing else, another type of sponges that have that beautiful lemon yellow stock. I don't know, what do you think, pilot? Any of those possibles in that little sediment sort of area there?
Look.
Sometimes get fooled. They can really look like they're loose, but they're not underneath.
These guys look pretty cemented.
Yeah, I would agree. Now that we're getting up closer.
Off to my port side, pilot.
That's copy.
It's worth occasionally looking down and seeing if anything looks like it might work. There's a dead sponge stock laying in amongst the rocks.
You can bump me in again on sonar. Yeah, if you would, please.
You got it going from 30 to 20.
Or something could have fallen from here.
There's a part of a pillow that kind of broke open probably shortly after it formed, and then the manganese crusts have coated the broken part of it.
Sounds good. Thank you, video.
And the term for those are broken pillows, believe it or not. So let's see if we've got John Smith. He's in the event log. He's probably monitoring passively. But now that we're into some of this geology, I want to see if we can coerce John into calling in every once in a while. Chris Ma is back right now, and so you can hear him talk a bit and track some of the requests via the chat room. Video. Oh, look at that. Perfect timing, Chris. Well, no, we had another crinoid, excuse me, as well as his second crinoid.
Okay. So it looks like we have a feather star or a crinoid. These are echinoderms, and they hold their arms up into the water. They have little feathery projections which come off of them. They're able to pick small organic food out of the water via these kind of filters that they use on their arms. That organic food is then processed and moved down to the mouth, which is located right in the center of... Holy cow, what's that? Was that a snail in the center? Or is that... Huh. Okay, that might just be the anus of the animal extended. And a lot of filter feeding animals, it makes sense to sort of have the outcurrent vent, if you will, projected a little higher away from the mouth. The mouth is right at the center there, where those 5 lines converge in the middle of the disc. So that might just be something. There are some animals that's called an anal chimney, where the excrement and other waste is sort of safely removed away from the mouth. And you'll notice below it there are arms or structures called cirri, which the feather star uses to adhere to the bottom. A lot of these crinoids, these feather stars, can swim, and some have the appropriate connective tissue to actually, if it was properly provoked, to actually sort of move its arms up into the water column in a wave-like motion and swim gracefully into the water.
Are we...
Oh, and so yeah, thank you. We can move on.
Okay. Come on out, video. I don't have a stable hold here.
And there's a coral up to the right of it. If we could take a look at that, please.
Sure, watch. We're just going to do a quick change out.
Oh, I'm sorry. Still getting my bearings myself. Thank you.
Hey, Josh.
Hey, dude.
Up and Away is
coral.
So this is Chris Ma from the Smithsonian's National Museum of Natural History. I'm here with my colleague Chris Kelly from the University of Hawaii, Currently, we're having a watch change. So our pilots are currently switching out from the team that was here this morning to the next. We're about to move on after that's completed and look at some other things.
Hauling up.
This is the Okeanos Explorer coming to you from Johnston Atoll
15
in the
meters
Central Pacific. This
range,
is
serials to
the
the
Lima
wall.
Okamoana expedition.
We have a ship moving.
Vectors.
Looks like
we're looking
we're
at
about
a
done with that move.
big bumpy field of basalt, I think.
Yeah, let's get this coral video.
Yes.
Copy that.
When you're ready, let's take a quick look at this coral, which Tina Molitsova from the Shirshov Oceanographic Institute, I believe she's identified this as Pleurogorgia. And these are octocorals. When we've looked at these more closely in the past, we've seen that the feeding polyps, all of the little flowery structures, have 8 feeding arms around the mouth.
I'm going to try to creep in on it, video.
Copy that.
And so all of these feeding arms are extended on these extensions into the water column, and these are extended in order to catch food. These are like other cnidarians. They have stinging cells, so presumably these capture not just organic particles, but things that move on their own as well. I'll have to double check. And we also see a barnacle here. We'll get back to that. But barnacles are crustaceans, similar to the animals that, well, they're the same animals that you see if you're looking at the side of boats, or perhaps on the rocky intertidal, or sometimes people even eat them. This is that was a stocked barnacle. They has its feeding tentacles extended into, or feeding arms, or legs rather, extended into the water. But I think we're satisfied with this, so whenever...
Yeah, come on up video.
Thank you. And and I think we'll take a look at that coral on the upper side there as well.
Come on in video.
So I believe this is Chrysogorgia, if my shoreside folks can chime in. Looks like it has that sort of gold sheen on the stalk. Aha. And so Scott France from the University of Louisiana has chimed in and confirmed that this is indeed Chrysogorgia. The name translates to gold coral, a name it earns very well because the stalks on these animals are gold colored. Is that somebody on the line? Very well. You can see all of the little feeding tentacles opened up into the water column. There are 8 of them. This is, again, an octocoral. And we also see a commensal squat lobster sitting in the center of its branches. And Roland's sharp eye points out I think that's a little snail that's present on one of the branches. So this is typical of a lot of these deep sea corals. They form little communities in and of themselves. They're often referred to as ecosystem engineers because their physical structure forms a little microcosm of different organisms which live in and around the animal. Just, in fact, you can see there's a little of these pink worms that I think yes. So if we could zoom in on those if possible.
We're at max zoom.
Yeah, I'll come in on it.
Yeah. There are these tiny little polychaete worms that are also present on the branches. They're orange. Yeah, like that one down there. You can see the little legs coming off to the side. These are segmented worms. Oh, wow. Getting a nice close-up view of all of the feeding tentacles as we proceed to zoom in on the little polychaete worm hanging on there.
A little current pushing me off the wall here.
Oh, really? Okay.
Yeah. No, that's all right. Just talking to myself.
Good to know, though.
I know they just wanted the
you can see
amphipod.
the multitude of feeding arms extended from each of the polyps.
There's your work.
Okay, so Les Watling thinks that this is one that we have not seen in the Central Pacific. What does the chat room think of collection? In the meantime, we're also seeing more
Pilot,
of
if they have that
the
amphipod, I might pull
multitudes
out.
of animals that live on the surface of this animal, including looked like there was some kind of crustacean, which we can take a look at.
Let's come back in. What would you like to see, Chris?
There was a pink sort of an amphipod or crustacean or something that was in the center of the screen, sort of in the background.
Come on in, video.
It was right next to that worm that we were looking at earlier. To the left, I believe.
Okay.
Oh wait, maybe. Oh no, that's the worm again. Wow, there's like quite a few of those worms on this coral. There's three of them that I count.
There's something big up near the top of this.
Okay, so maybe wow, there's quite a few. Maybe I think the amphipod you were seeing were more of these polychaete worms living on the surface. Yeah, I think that's another one of these. Is that a polychaete worm or an amphipod? That's a polychaete. Yeah. Okay. So yeah, so this is interesting. There's these interesting little orangish, clear polychaete worms, and there's a nice one right there that are all over the surface. Okay, so waiting on one more person from shoreside to verify. So, but I think we might be wanting to collect this one.
Hey, come on out, video.
Co-pilot, how are you sitting if they want to go for this?
We're good.
Full wide.
All right, great.
We're
But
good.
yeah, I think it would be
Inching ever so closer, but we're still
Full
12
wide?
meters off.
Yeah.
waiting for Scott France to get some verification if they want to collect. So if we could get into position as a whatever
You
pre-preparation you need.
All boxes available.
Copy that.
Thank you. And what's this thing on the left?
We can take a look
This
at it.
brown. Yes, please. I'm sorry. If we could.
Right, let's get the guy on the left. Come on.
That sort of. Oh wow. Okay. That's an unusual 5-armed crinoid. That's a feather star. I know that's something that we've seen before, but I'll have to look it up. But it's very unusual for crinoids to have just 5 arms like that. This really shows off their pentameral symmetry. This is a kind of echinoderm. This is a group known for having 5-part symmetry. That is, they have exactly their body symmetry matches into exactly 5 parts. But the arms come off, and there are projections that they extend out into the water, which they use to capture organic particles as they float by. And it looks, again, like you can see that little bulb there. I think that's where the excrement is projected out from the disc after feeding happens. And then there's a mouth at the center point where all of the 5 lines converge
hydraulic
in the
pressure
center of the
feedback
disc.
on D2. Is
Okay, so we have 2 votes from both Scott France and Les Watling to
I saw
collect
it turned
the
off. The data looks
Chrysogorgia
kinda
that we were just
junko.
looking at.
Yeah,
So,
it's,
pilot,
not-
at your convenience
I'm not sure
and
if it was
at your
entirely
pleasure,
removed, but
if we
it's,
could ask you
it's
to
useless.
collect, please.
Okay.
So you
Roger
just
that.
Max
for the
the
craft, not necessarily max, but maybe 2,000 RPM, something like that, right?
1,000 might be about as all we've got.
Okay. Maybe I'm off on that number.
Okay, well, we'll just, that should be good.
3/4 is probably good.
Happy about that. Starboard about.
And so what's happening now, for those of you who are just tuning in, this is Chris Ma from the National Museum of Natural History at the Smithsonian. And I'm here with my colleague Chris Kelly, who's currently away at lunch. He's from the University of Hawaii.
Hydraulics are up, and I'm assuming
NOAA Okeanos Explorer Expedition. We're currently getting set up for a collection. We're going to sample this Chrysogorgia coral, this golden coral.
Let me get you.
With this many
get
interesting
warmed up there. I'll get port bio
different
out.
forms living on it. There's a there's a bunch of several polychaete worms living on it, as well as I believe that crab that was in the center, or squat lobster rather. So the pilots have extended a hydraulic arm, which we'll use to grab the specimen and transport it to a special specimen box, which will keep it in water in the ambient temperature in which it was collected.
So we want the whole thing if we can get it?
Yes, please. Thank you.
Okay, copy that.
What's going on?
It's wiry, if that helps.
Roger.
Drawer coming out. I'll leave you a little bit of room to maneuver there before I extend more.
Yeah, copy that.
Nice grab.
Oh, beautiful. Thank you.
Okay, continuing to extend.
Go for it. Nope.
Drawer out. Oh, did I bump you off the cliff? You're probably all right.
I just don't want to. I don't want to bang up the drawer here.
Okay.
I'm just gonna pull up and
float off.
Go into auto depth.
Okay.
So which box will that go in?
Not sure just yet. It's going to be in the port side.
Very good. Port side bio box, presumably then.
It's a little big.
I'd have to pack
So
it
for
in.
those who are watching, this is Okeanos Explorer. We're currently collecting a gold coral, which is potentially a new species. Our pilots have very deftly collected a coral with our hydraulic manipulator arm, and they're currently trying to get it into the sample box for a sampling so that we can examine and study the specimen for later. There's a great deal of skill that's involved with this kind of work.
All right, clear.
Gently.
And even with the advanced electronics that we have, there's a great deal of concentration as well as hand-eye agility that goes into collection.
Retract complete.
Chris, as a bonus, I think that crab was included in that collection. I didn't see him leave.
Yeah, it didn't seem to run off when we
So
collected
I have
it either.
sort of gone a little bit northwest of you and around the corner here.
Scoot to my starboard. So that went in the port inner, Chris.
Okay, so it went into the port inner.
Let me
you
haul
got
up
that?
a little
okay.
bit too.
Thank you, pilot.
You're welcome.
Spooling down the HPU.
All right. So that's stowed away. Very good. So
so we've been moving.
Now is probably as good a time as any to also let the forward engineering crew introduce themselves after they've successfully completed this great specimen collection. So if you guys have a moment, if you could introduce yourselves.
All right. So this is Joshua Carlson. I'm piloting D2 at the moment. And to my right is
Dave Wright.
Dave Wright. He's piloting Sirius. To my left,
Dave Casagrande, navigator.
And on video,
Tara video in the video clipping chair.
And Tara is in the video clipping chair, and we're all engineers and videographers who work for the Global Foundation for Ocean Exploration.
Thank you. And this is Chris Ma from the Smithsonian's National Museum of Natural History. This is the Okeanos Explorer. We're currently exploring an unnamed seamount in the Johnston Atoll region. We're currently at about 2563 meters, and we're currently surveying
looking at
sort of a basalt bottom.
75 degrees down.
Roger. I'm going to push ahead.
And we were moving at 0200, steer off a bit to port from there, unless we want to.
So we can proceed
as swung my head quite a bit to
for whatever plan is established.
Keep you in view. I'll follow you.
The final waypoint.
Yeah.
Is just about due north from us now.
There's a holothurian on the right.
But I think we can follow local up.
Oh, is that a sea cucumber on the right side, that purple thing?
Yeah. So I'm thinking I see a loose rock here. I'm just going to poke it with my skid real quick.
Yes, a loose rock would be good if we can get it. A lot of it looks like it might be ideal, if it's the right kind.
Oh, there's some loose rocks.
Yeah, it moved. I can see it in bubble there. Spooling up HPU.
So both of these are loose, which the lasers are on right now.
Okay.
I don't know if they're of the shape and size you want, but either of these are loose if you want to grab it.
Sure. Let me take a look. I think. Sure, if these are pillow lavas, I think. Let's take a quick. Is there a quick way to just sample whether or not they're loose?
Yeah, these are loose.
Okay.
Yep, I pumped them with the ROV.
Okay. Oh well. All right. We might as well get one now then.
Okay.
Yeah, port rock box probably be simple, and I won't shove you off. So hydraulics are good.
Thank you.
Let me just sit down here.
You might straddle that big rock.
Yeah, I just want to be able to move the rock box inboard.
Ah.
So if I straddle it, then
yeah,
I can.
right. Okay. Forgot about that aspect. Yeah. Okay.
Descend just a little bit, Dave, or are you too close to the...
I can come down.
Yeah, I think I'm getting pulled a little
bit. Sure. Yeah, I was hauling up in anticipation of a departure there. That should make you happy. Let's see.
So we are now engaged in a geological collection. We're using the manipulator arm to collect a rock sample to help us understand the geology, the geological history and composition of this seamount.
Do you want to open that box first?
Nah.
Yeah, well, I want to squeeze it just.
Okay. Do it with the rock. Port wing fully inboard.
Is there a preference on which rock, or is either one fine, Chris?
So, Chris, can you provide input as to which rock is generally preferred?
That one looks a little skinny and flat. Maybe that thicker one would be better if it'll fit.
I think it will.
Yeah.
You thinking it's mostly crust?
Yeah, that's what I'm a little worried about. I think it is too. Yeah. Maybe you ought to
try whacking it and see if it busts open.
So what happens sometimes is a crust breaks off and then rolls downhill, and that's what's loose. But really, the geologists are particularly interested in getting the salt.
So we want to pass on this, maybe?
You know what? It's in the manipulator. We could throw it in the box, and maybe we'll see something better. But having one in the box is always a good idea.
We can always take it out later if we need the room.
That's going to be port forward rock box. That's what he's putting it in right now. And I would be a little bit reluctant to call it basalt. I would just call it manganese crusted rock or something like that.
All right, you can close that.
Great, thank you, pilot.
We'll,
the lid should close.
Ah, how convenient.
The auto lid closer.
Auto magically, eh? Up, other way. Bueno.
Thank you.
You got the waypoint?
Yep, I got it.
All right.
Onwards and upwards.
Onwards and upwards.
HPU secure.
So the way these things work is now we'll see the perfect rock.
That's fine. We'll get that one too.
Any interest in these sponges here, Chris?
Or? No, Josh. We got these are bolosoma, and I think they're pretty much the same, except maybe this little white one that's over closer to the ground here might be something different, but I'm not sure.
Come on in video. Just do a snap
maybe a
here.
snap zoom, yeah.
Oh, what's?
Yeah, I'm kind of
dead.
I don't know what we're looking at here. Is that sponge part of the? No, it's not, right? Or is it?
I'm not sure.
What is going on here?
So I think it's a good bet. We're going to spend a few more moments looking at this, pilot. Thank you.
There's also some rocks right here too that look loose.
Oh yeah, these look better. In fact, the one right next to the sponge looks reasonable. I got it. So this is one of these big bolosoma. The base is to the right, and the top ball used to be to the left where the lasers are, and that's the remnants of the ball itself.
Oh, interesting.
But on the other hand, look at that rock. That one that's right is.
So that one with the lasers is too big.
Yeah. How about the one behind it?
That one is too big.
Oh.
This guy is.
That squarish one right by the stem?
How about that one?
Yeah, that one we can do.
It's got angles like it could be.
More recently
could
removed.
Be okay.
There's even one above the stem that looks. Almost looks like something got knocked loose from above. Back on hydraulics.
I missed that. They want this?
They're thinking about it.
Okay.
So there's this guy here.
Yeah.
And then there's this guy here.
Let's look at that one there, that second one. That looks nice.
Right.
Any chance we can dump the first one and put that one in instead?
We can try.
We have more boxes.
We can take them
both, only
can't
just
we?
what, 10 feet or so away from where we grabbed it. Yeah, that's definitely a better rock.
Do you want to take both, or do you want to try to drop the other
let's
one?
take both.
Right.
Yeah, I guess that makes.
Let's do starboard.
Wing inboard.
You never do know.
Do you want to zoom in on this, or you?
Starboard rock
yes,
box.
please, if you could.
Yeah. All right, zoom in video. Starboard rock box.
Starboard wing coming inboard.
So some of you might be asking, why is this a better rock than the other one? And what we are looking for basalt. Sometimes the manganese crust can form up to 5 centimeters in thickness in this particular type of seamount, or on this particular type of seamount. We try to find rocks that are kind of angular and square that might indicate that they really are basalt, not all just manganese crust. Because if they've got basalt, then they can actually use that rock to do some geochemistry and figure out what the age of the seamount is, as well as a lot of other very, very useful information. So we do look for these angular ones. We hadn't seen this initially, but I think this has got a chance of being a really nice rock for the geologist.
Okay. You happy with that?
Yeah, I am happy.
Still this guy.
Yeah, let's keep it as a generic.
If you can swing the arm outboard when you stow it,
that's going to go in
less
the
of a
starboard
shadow
rock
in the
box,
sonar.
rock box. That's it.
No, copy. All right, we can still the starboard rock box.
Starboard wing, going outboard.
What is going on here?
All right, we got the waypoint.
Fastest
Okay.
samples.
Are we all set?
Marked.
Hang on for just a second, please.
Holding.
Want that too.
Both of us are having some computer issues here. My laptop is wigging out. And
That never happens
Nicola's
up here.
her own.
Do you want to pull up the sample
Nicola
page?
is actually punching in the collection
I don't
data,
actually
which
know
is
how to do
really
that.
crucial.
But I could.
Sampling. You're good to go? Okay, we're good.
All
Thank
right.
you. Thank you, pilot.
So if they ever need you to pull it up on one of these computers, it's 192.168.4.201/ROV sampling. ROV-sampling. All right.
Helm ahead full.
Onward and upward.
Got 2 rocks. One of them could be really good. There's another, again, where you mentioned these beautiful bolosomas. Yeah.
ROV-sampling. So, should you ever need to,
Sure, I'll bookmark this.
as a last resort. Also, if we have the waypoint, we can pull out the data afterwards
Sure.
from the CSV files. So.
Good to know.
So I'm looking northwest, and 56 degrees, delta 17.
Copy. So, what's the bearing we want to be heading on?
It's just about due north.
And
I'm gonna
I'll bring my head
watch
north
this
here.
data.
Copy that.
Watch lead nav.
Roger, Nav, go ahead.
Just a little bit of an update on how we're doing. We've covered 315 meters. We have 500 meters left on this track to our final waypoint. And in order to have time for our mid-water work, we're going to need to leave the bottom in about 3 hours, 15 minutes.
Okay, thank you very much, Nav, for the update. So I guess we'll, I guess we'll just try to keep the pace up as we go along, and we'll try to, focus the close-ups on things we haven't yet imaged, if we can.
Sure.
All right, we're in a good arrangement here if we want to start moving. I always forget to adjust my Z-bias after I pick up rocks.
It's kind of like the heavy feeling we get after lunch.
Exactly.
Can we adjust our Z bias?
That's what the espresso machine is for.
Right, of course.
Pilot bias adjustment.
Co-pilot, whenever you are ready for a move, let me know.
Co-pilot is ready. I'm showing us 25 meters off the wall. There's a little bit of stuff to my left, but basically you can bang in 1 or 2 tether lengths here and we'll be happy.
All right, I'll do 30 meters.
Snap in here video.
At due north.
Oh, here we go. You're on, Chris.
So we're coming on here as a beautiful purple sea cucumber. These are these are the same kinds of animals that you see intertidally or in shallow waters. But in the deep sea cucumbers take on a completely bizarre appearance. This one, for example, is not only purple, but is actually somewhat translucent. And what you're seeing through the body wall is actually its gut. So what these animals eat are sediment and organic particles, and they digest that through a mouth, which is probably right around that deep purple fringe near the head. And so it's absorbing those sediments and organic particles into its gut with feeding arms. And so what you're seeing is the food in its gut literally moving through the animal as it then excretes them out towards the rear end. So it's sort of just a big mouth with the processing center in the middle and the exit point towards the end. This one looks like it might be a synalectid in the genus Hansenothuria, possibly. A lot of the identifications of these animals is somewhat complicated because the structures are not always clear, and even the original material is often badly damaged during collection.
Thank you, Bridge.
But as you can see here, we're looking literally right through its body wall into its intestine. And I can identify a loop. There's also some structures sort of more meandering towards the end there. But some of these, as we'll see, actually can swim. So thank you very much, pilot. We can move on.
You're welcome. Come on out. When you're clear, video.
Yeah, just another second.
Yeah, Roger.
Specimen view.
Roger.
Sort of a younger white Bolosoma sponge there next to it.
Is that a new code? Specimen U?
That,
specimen.
when they're
what,
really
what did she say?
small, they aren't lemon yellow.
It's like a specimen shot.
That's interesting. Yeah, it could change color.
Oh, specimen view.
Yeah.
Yes, got it.
Again, one of the mysteries we haven't really cleared up.
For the animal guide. Like to get a full frame shot with lasers. It's a good.
What you're seeing is the deep sea bottom, sort of a very amazing. I'm not sure if it's basalt, but a cemented surface with lots of boulders and other rocks.
This is definitely basalt.
It's basalt.
It's pillow lava formation.
Okay.
This is what we're seeing, but very heavily manganese crusted.
So it's both.
That was amazing that Josh was able to find a rock there that looked like it's pretty good.
Lovely. And there's a coral in the middle.
Yeah, this is Pleurogorgia. We could probably. I think it's Pleurogorgia. We could do a snap zoom on it just to confirm.
The one in the back or the one right up?
Which one in the back?
There's this big bushy one in the back, and then there's this whip-like thing in the front.
The one in the back is Pleurogorgia, I think. I think that could be a dead sponge.
Yeah, might be a dead. Let's do a snap on the one in the back.
Yeah.
Pleurogorgia.
That's also
militaris.
Yeah. To the left, there's a snail on the rock, as well as a brasingid, which we could take a look at for a few moments. But the snail would be really interesting, I think.
We need to get closer to it.
Yeah.
Want me to go wide?
Yeah, come out wide.
Video swapping back out.
Yeah, do I have enough leash to get here, Dave?
Just a bit. I'm gonna come
that way.
I'm coming down.
Okay.
Whoever picked the new chairs, the recline is nice here. You need footrests though.
Yeah.
I got a footrest I could slide down there for you.
Well, it's there's
Come on in, video.
stuff in the rack in the way.
You feel there's a little squeeze thing on the side?
Yeah, I told them
The
about
lumbars,
that one.
yeah.
That's nice. Much better than the old ones. All right, I'm stable. You can go as tight as you want. Video.
Interesting. So this is a snail like the type of snail you would find in your backyard. but a very different, probably family, genus, and species. They are probably there are any number of things they could be feeding on, including the organics, or possibly even some of the corals or other animals around here. We don't see a lot of records for small animals like this, so I just wanted to grab this one just for that. So let's move on.
Yeah, come on out, Mike.
Yeah. We can actually skip the Brisingid in hindsight, so
Okay,
we can just move on from here. Thank you.
Do a snap here. Oh yeah, what's that? Come on tight.
Sponge, I think.
Oh, look at that. Sorry, I've been trying to get my laptop computer going again. I had a little technical issue. I was looking down after restarting the computer. This is, I believe it's a demo sponge.
Thank you, Bridge.
In the genus Piloderma. These are like toothbrush sponges is what we called them in the past. And I think
Is that move complete already?
Yes, it's just ended.
I put in another one.
In the Johnston Atoll
I can look
area.
back at him if I overshoot him,
Of
but
them, so we may see a lot more of these
Okay.
as we move up slope.
Get us
just
get
in
us
the
going
interest.
again. Sure.
Yeah.
Let's get a tight shot.
I'm gonna just double check the ID here in our Okeanos animal log. That way, there's an online
Bridge,
version
nav.
of that animal log. There's an online version. Those of you who are interested, it's being improved every single year, trying to make it more user-friendly.
We'd like to repeat that.
Yeah,
last
so
move. That's
I'm pretty
30
sure this
meters,
is
bearing 000,
a demo
speed 0.2
sponge. It's not a
knots.
glass sponge. It's a little bit different group of sponges. And genus is Piloderma. It's in the family Dendrorhizelidae, and I'm not very knowledgeable about this group. In the Marianas, we had Shirley Pomponi, who was on board for that, and she specializes in them. So I think Shirley's on travel now. But anyway, I think that's a pretty good bet. Pretty good guess for that one. Thank you, pilot.
Come on out. See the current kicking out. Little vortices.
You've got quite a bit of tether and more coming.
Right.
I'm starting to really move now, so you can
Thank you,
push
Bridge.
ahead
Copy.
a fair bit there and maybe get into some more interesting territory.
The nodules are bigger here.
It's another
in addition to
Pleurogorgia.
the cemented background.
Sort of a wispy one. Still a little sparse. I was kind of hoping for a few more animals, but maybe again as we get a little further up slope, we'll get into a little higher density. Got a little ways to go. We've covered a little over 300 meters, and we usually try to, if possible, get to 800 meters distance in a dive. So we got about 500 meters to go, and we're going to be leaving a little bit early today to accommodate the midwater. So we're going to try to go at a reasonable pace. Of course, if we find all types of amazing things, we'll pause and take a look. And that is a, I don't know, dead stump or something, I think.
Let's do a snap on this.
Yep. It's a dead holdfast, maybe of a sponge. Not really sure.
Come on up.
I think.
And there's another one of the little toothbrush sponges.
Yeah, well, they may start appearing in the hundreds and then thousands, and then it gets crazy.
We're starting to get into the Carl Sagan magnitude.
Yes,
millions
exactly.
Upon millions.
Yeah.
Billions and billions.
Okay, I'm back on the event log finally. I see Scott concurs. Scott France of University of Louisiana at Lafayette. He was on that cruise in 2015 down here to Johnston, and he concurs that that's when we did see a lot of these toothbrush sponges. So that's great. All right, we've got quite a lot of people now in the event log. We've got a bunch of the midwater guys who are joining early as well. Dougal Lindsay, who's from Jamstec, he's here eagerly awaiting the midwater portion of the dive, which will occur a little while later. And Mike Vecchione, they're at the Smithsonian, and also Noah OER. He's another midwater specialist. And maybe we'll see some jellies or some things floating by for those guys. We've also got Tracy Sutton joining us as well. And so we've got a lot of specialists online. Give us a hand today.
Let's do a snap right here.
Yeah, another of these toothbrush sponges, the Piloderma. It's got a little gap in the middle of it. If something was munching on it, maybe one of your evil beasts, Chris. I don't know.
I would love to see one at this point.
Yeah, come on up.
Yeah, we have not seen, except for that Brazingid, right?
Yeah.
That was it.
What's that white thing?
So, co-pilot, that DC bus return is up to 850K.
All right, come on in, video.
They're working on that camera, so I think pretty happy with that.
Okay.
Yeah.
So I had. Oh wow, this is kind of neat. Oh, okay, there's like a bunch of different things here. So this is a feather star, which is perched on, I think it's a dead Chrysogorgia stalk. So crinoids, as I'd mentioned earlier, also called feather stars, hold their arms up into the water and can pick food out with the little projections on the arms. Also taking advantage of whatever current is flowing at this height above the bottom is this really attractive looking barnacle. This is a stocked barnacle, so barnacles that you might be familiar with in shallow water are more like acorn barnacles. They're kind of like a flat cup of plates. But there are other types that have these thick, fleshy stalks. And in the deep sea, there are these very big and sort of ancient-looking barnacles that this one has its feeding legs extended, very handsome ones, I might add. And it looks like it even has a little barnacle growing off of its stalk, off of the stalk itself. So most people don't realize that not only are there barnacles, looks like we scared it a little. Most people don't realize that not only are there barnacles, commonly on shore near rocks and on boats, but also in the deep sea, as this one is at about 2,500 meters. As we skim down the surface of this dead coral stalk, we also see lots of little animals growing on the surface, so those might be hydroids or other types of things. So every kind of substratum, every kind of surface that presents itself in this habitat becomes a home for something else. In this case, these animals are able to exploit the currents as well as the space that is present. So anyway, thank you, pilot. Let's continue.
Left on
Come on up, video.
Yeah. Well, it looks like the ship's starting to.
Yeah, it is.
Just got the little hook there at the end.
It's That's the direction the wind and currents are moving us, but looks like it's stopped.
Okay.
But it comes back.
So you got about 20 meters of leash in front of you there, Josh.
Copy that. Is this something new?
No, I believe that's the Chrysogorgia we collected earlier. We can move on.
Copy that.
Thank you. And that's another Pleurogorgia, just noting that for the record. Looks like another crinoid or something. Maybe it's a Brisingid. We can move on.
Let's do a snap.
I think the crinoids I think that's the same crinoid as we saw before.
Thank you, Bridge.
Oh, it's one of those weird little. Yeah. No, but there's this little yellow stalk thing that looks like one of your bizarre bryozoa things. Very good, pilot. Thank you.
So when we were stationary for a while, he settled out pretty much right under the stern.
All right. So where is
I'd say you
our
have maybe 25, 30
amazing
meters left
creature that's lurking behind one of these rocks, just waiting to be discovered?
The next big thing.
Look up ahead of me just to make sure there's nothing scary.
A little close, but
look at how
Is there a
round.
little white thing on the
I
rock
operate
just in lower
closer
center
to
screen
you
there?
than others
Not the
do.
coral, but to the left.
Just to make
not
you look
seeing
good.
what you're talking about.
You're just
Chris.
you're right above it on the lasers.
Oh, okay. Come on in, video.
Oh, it's a little bit of junky stuff.
All right.
Sorry.
No, that's all right. Snap zooms take no time, and they give you guys a better idea of what we're seeing. So
thank you.
I got to push ahead a little bit, getting a little bit behind.
Yeah. One of those things I've always wanted to say. Head full. Head full helm about a head full.
That's certainly not one of the things I always wanted to say. What I always wanted to say is, woohoo, won the lottery.
Well, yes, but different kind of dream, I guess.
I guess so.
I'll have to say that later in my Captain Picard voice.
Right, so there's probably some interest in this elongate whip that's right in front of us. I think it's a bamboo. I don't know if it's the same one as we've seen before, but I wonder if we could do a snap zoom on it.
Yeah, right.
Oh, wow.
Yeah.
Right, come on in, video. I'll approach. Get some light on it.
It's about our height, isn't it?
Yeah, at least a couple meters.
So what we're looking at is a bamboo coral, and it's an kind of octocoral. And so what we're looking at here are the various feeding polyps, the part of this colonial animal which feeds. And so off of each mouth that you're seeing, each one of these sort of orange dots are 8 feeding arms. Each one of those arms has a small projection that's coming off of it, which is what helps us to identify what they are. But the animal's colonial, and so the tissue that you're seeing is all part of essentially one animal, and then they're held by a structural support down the center of that stalk, and that structural component is both white and with dark bands, and that's what earns the animal its common name, bamboo coral, because they look just like bamboo.
All right, so I'm waiting to get some commentary on this species. This is not the same bamboo coral as we imaged earlier, at least before I went to lunch. Does not have those very long spines coming between the tentacles. This is a different beast. And I know that Scott's on. I don't see Les at the moment. So Scott, can you give us a hand on the idea in this one, at least going to clade? you can go that far. Hardly any of the sclerites that are in the column itself, which is definitely informative. I don't see really hardly any intertentacular spines also. Now, one of the keys to identifying bamboo corals is whether or not they're branching from the nodes, that black area, and or the internode, the whiter areas that we're running through. And the problem here is that for these unbranched bamboos, you don't have any of that information. There's no branch points. So Scott believes that this could be in the Jasonisis clade, so it just shows you the variability that he and Les and the others working in this group, Abby, are having to deal with. The Jasonisis that we saw yesterday was this beautiful fan, and today we're seeing an unbranched form. It's in that group. That doesn't necessarily mean it's necessarily in that genus, but anyway, thank you very much, Scott. He noted that they're very short rod-like sclerites that you can occasionally see in the polyp wall, and these are kind of the clues that can lead to an ID, and that's why this video is so amazing, that you can actually see microscopic parts in these rooms. Pilot, we're good whenever you are and video is.
All right, come on up, video, when you're good.
So now we're looking at 6 minutes since the move ended, and I'm just slowing down now.
But basically say for Jasonisis, the genus itself, he would have expected more scales and a little bit milkier looking tissue. So could be something related, but not necessarily in that genus. Thanks, Scott.
Quick snap here. It's a sponge.
Looks like we've got a small ferronematid sponge. I'm not sure it's Poliopogon or not, but I believe it's in the family Ferronematidae. It's a glass sponge. And again, what I'm looking at is how it's attached to the rock, which is it's got a fibrous attachment. So that helps a lot. It's got sort of a thicker columnar stalk on it, which some of the ferronematids have. So that's great. Thank you very much.
Okay, come on up, please.
It's on here.
There's a lot of potentially loose rocks here, but they're all kind of rounded, and they kind of have, well, looks, probably a lot of these are just pure crust. I think that second one we got is a pretty decent rock. It'll be interesting to see what it looks like when it gets on the deck.
Let's do a snap right here. Any interest in this shrimp?
Sure. I think it's going to be a nematocarcinus. I think it's the first one we've seen.
Video, can we get a zoom on the shrimp?
We don't need tons of time on it since these are pretty common. You've entered some before, but yes, it is. A long-legged shrimp, named that for obvious reasons. This is in the family Nematocarcinidae and probably in the genus Nematocarcinus.
Co-pilot.
These are this group of shrimp.
Yeah, I'd call it in. Get us
everywhere we've been in the Pacific, from the Mariana Trench to Wake to American Samoa, Howland and Baker, Hawaii, and Johnson. So they're a very successful group down here.
Bridge, nav.
Yeah, come on up, video.
Another ship move, please. 30 meters, bearing 000, speed 0.2. Good copy.
This is a little different sponge. If we could do a snap zoom on this.
Copy
This
that.
is not the same stock sponge that we've been seeing all day, which we've been calling bolosoma. So this is good. I was wondering when some of these would actually show up.
All right, come on in video.
So this is yet another form of glass sponge. So we're seeing a variety of forms here. In this case, it's in the family Roselidae.
Thank you,
It
Rich.
is a stock sponge, obviously. And in the genus here is Caulophacus. And we saw one of those, or a couple of those yesterday. They're beautiful sponges. They're being worked up right now because most of the ones out here are undescribed species. So yeah, thank you very much, pilot. Whenever video's ready.
When you're good, video. Quick snap zoom here.
This is a smaller unbranched bamboo coral, smaller colony. This, I think this is yet another different one. All right, Scott, if you're making dinner or you're doing something, you got to get to your TV or your computer. I need some help here. The Scott France spotlight shining up against the clouds right now.
Yeah, if you have any more zoom. Where's that max?
That's max. Different from the previous Bathygorgia.
So did you guys image a Bathygorgia already? Maybe when I was on...
I mean, he was just saying.
Oh. So yeah, he thinks it's different than the first whip he saw, and he thinks it's potentially in the genus Bathygorgia. We've seen a number of those during the cruise, they're suspected in that genus. I think we've collected a few as well. Great, thank you very much, pilot.
Okay, when you're good, video. Stocked crinoid, or?
Yeah, let's take a quick look at the stock crinoid.
Come
just
on in, video.
Snap zoom.
Yeah, there we are.
Yeah.
Okay.
So this is a. So we've seen other crinoids. This is those are feather stars. This one is a stocked crinoid. It has a full stock, and look at that. There's a snail on the.
Wait, Chris, you're missing the thing here. It's
got
I know. 4 arms.
Yes, I see that. That's probably just an aberrant number of arms because one's damaged, but there's a snail on the anal vent. That could be a eulimid practicing coprophagy, or it could be like a sort of a vampire snail feeding on its bodily fluids.
So you think the snail is on the broken arm, and maybe the snail is that what? Is that
Oh,
where
it's on the
broken
arm. It's not on the vent, on the anal vent. Yeah, it might be on the arm. Just we actually saw a snail like this on the other crinoid we saw the other day. So that might be why. That might be what it's doing, is feeding on the fluids coming off of the body.
So is this a mutant crinoid? I mean,
No,
the fact that the 4 branches are coming out opposite each other rather
So-
than like
Right.
as though there's a fifth and one, there's a gap
right.
where one's missing.
So there's actually, I think there are 5 arms here. 1, 2, 3, 4 that are completely formed, but that fifth one, the one the snail is on, it looks like it's damaged. So either a fish took it off, or some other kind of predator took it off, or it's possible that the snail might actually be contributing to the fact that the arm isn't completely grown. Either way, it's a fairly interesting observation because the effect of these parasites or these commensals on these crinoids is really poorly known. So this might contribute to the number of arm variation, the amount of arm variation that we see in them. The ecology of these things and their habitats is also really poorly known. So this is very novel for that reason alone. But the snail and the on the crinoid, I think that's been observed before, but this association with the damaged arm might be more interesting.
All right, so we've got a question from shore here from Derek Sutcliffe, who's at the Interspace Center. And this is for the pilot. So, Josh, you want to take this one? He basically wants to know from his audience there, how are you able to keep the camera steady and take such good video?
So, in this case, I set the vehicle down on the bottom. So I'm giving a downward thrust and a forward thrust in my toe. We call it the toes, but there's two parts of the frame that stick out, and I'm just, putting my toes down, and that helps stabilize everything. And then as I rotate up and down, this motion here there's a rotator on the actual camera. So I can give it just a little bit of an adjustment to keep it in view, and just adjust my my rotation just a little bit to keep it centered. So this is one technique we use, which is to just set down on the bottom, and then you don't have to worry about currents going around. The other technique is when you don't have the opportunity to sit down on the bottom, when, say, there's a lot of coral or a lot of delicate, old, pretty things you don't want to crush, you have the opportunity to sit down on the bottom. You can either try to be your own control loop and keep it perfectly centered, or what some pilots will do is let the vehicle drift in such a way that it creates a nice picture and keeps the creature in zoom. So, for example, right now I'm not on the bottom, I'm just floating, and the vehicle's so big that it's not going to move very quickly. So I'm just putting in small little adjustments, a little pulse of a thruster here or there, just to keep it nice and centered. And it's actually easier to keep the vehicle moving than to try to keep it in one spot. And because what will happen is you'll get these, pilot-induced oscillations where you keep trying to keep it centered, and you overcompensate, and then try to reverse it and overcompensate again, and you end up not getting a good shot at all. So, yeah, those are just some of the techniques we use to get these nice shots.
Great, thank you, pilot. And as you're pulling up here, you're a different group than the group that started out this dive. Did you guys introduce yourselves already or not?
Yes, we did.
Oh, you did? Okay.
We can come by
lunch.
Oh, yeah, I did that right after they
switched out?
Yeah.
Oh, okay.
Right over you.
All right, pushing ahead.
And the ship is done moving, but as we found, about 6-7 minutes left, probably.
So our current depth is 2,537 meters. If you're just joining us, we are on a seamount that's a little ways north, probably about 125 miles north of Johnston Atoll, in the Johnston Atoll unit of the Pacific Remote Islands Marine National Monument, one of the marvelous marine monuments we have out here in the Pacific. And we are surveying this seamount, which has never before been explored. There's never before been any work on this location, and we are seeing it for the first time live. And as you can see, there's some marvelous life that exists down here, well over a mile below the surface, and very cold temperatures right now. The temperature is 1.8 degrees Celsius, just above freezing, and our pressure is extremely high. Our oxygen level is about 2.4 at the moment, so very low oxygen, at least for we humans. There are other animals that can do quite well in those low oxygen values. At the surface, typically the oxygen in water is about 6 to 7 milligrams per milliliter, and we're now at around 2, so we're about a third at least of.
Bob, let's just do a snap here.
Probably a little bit more than a third, but the oxygen levels are at the surface.
Oh, so what do we got here? Some kind of discoloration, I think.
Yeah.
Huh.
Type of a mat or something used to be there.
Yeah.
It no longer is here.
Right,
Broadcast is coming from the NOAA ship Okeanos Explorer and their remotely operated vehicle, the Deep Discoverer. It's a chrysogorgid coral in front of us and another bamboo in the background. Looks similar to the one we imaged, that really tall one, a few minutes ago.
Is something different?
Yeah, no, I was just about to comment on that. I'm not positive. It could be.
Right, come on in video.
Could be just a thicker version of those toothbrush sponges. I'm not sure.
Just come on in video.
Okeanos.
Interesting texture.
Yeah, so. Well, I think it's a demasponge for sure, and it's either a kind of a slightly not quite columnar piloderma, or it could be something that we call Stellodorix, a different demasponge, but I think we'll just stick with demasponge right now. Yeah, thank you, pilot.
All right, come on out, video.
So, pilot, do you see all of those sort of white things on the side of the rock, kind of like its armpit in the front there?
Up by the lasers.
There's a curve in the rock, a convexity, and it's really, there's all these little white, they look like pods.
Down here.
Yeah.
Yeah.
I think it's just flocculence, but maybe it's something more.
All right, copy that. Come on in video.
Oh, wow. There's all these little
forams or something, aren't they?
Are they?
Well, I think we've defaulted to that in the past when I see these little bushy structures.
Huh, interesting.
Yeah, let's see if
so they're a mystery.
Yeah, I'm waiting to see if anybody in the event log can take a stab at these things. Come on, be brave out there. Scott France was brave. He rose to the challenge. He's suggesting they're arborescent forams, which I think is what we've been.
Yeah. So, so sometimes what we see in the deep isn't even an animal. Sometimes they're single-celled animals or single-celled organisms, and sometimes they create these interesting structures. This looks like some kind of. When they say arborescent, they mean tree-like, so this is formed as kind of skeleton that looks like a branching tree. And we have agreement from Mike Vecchione, but yeah, sometimes forams, little shelled amoebas can create these large, hard part type structures.
Move, please.
Looks like they're exploiting the habitat on the underside of this basaltic rock.
0 degrees, speed
anyway,
0.2
we can move on. Thank you.
All right, come on out, video.
Good copy.
Oh, that's a big break right there.
Yeah, look at the size of that. It's a broken pillow, I think.
Chunk of broken lava rock.
Yeah, pillow lava.
Probably date that.
Will that fit in the box, Josh?
Yeah,
I guess not.
I mean, we could try.
Could we just kind of attach it to the top with a bungee cord or something?
We'd have to pull D2 up.
I don't think we would float.
But it's definitely basalt, so that would be a great sample.
Did we bring the ratchet straps with us on this trip?
I don't think so.
Another two more Pleurogorgias. We've already imaged those today.
Should put a claw or Sirius. That'd be hard to do the ship motion.
We don't have a lift through tether attachment.
No, you need a lift through, yeah.
So, Josh, I don't know if you remember this, but in 2015, we collected a rock that weighed in at 154 pounds.
154 pounds?
Yeah, I'm pretty sure that's right. And
wow.
And I can't remember if that's the one that Daniel and I put in this big heavy-duty bin and muscled upstairs to the lab together. I will double-check that figure, but let's put it this way. It was big, and it was very heavy.
That was probably the biggest rock we've ever grabbed.
It was a monster. We couldn't find... It was almost loose in the bin when we shipped it to the Oregon State University because we couldn't find any bag or anything big enough to hold it.
I bet D2 was heavy.
It was.
Yeah, when we came up.
Yep.
Right, come on in, video.
Have a nice mushroom coral here.
You answered the question. That was probably the biggest rock you've ever collected.
Oh, that's so beautiful.
Yeah. Is Tina on? I think she is. Right, Tina?
So we draw upon the considerable skills of Tina Molosova from the Shirshov Oceanological Institute. This is her forte, various deep-sea cnidarians, animals with stinging cells related to jellyfish, sea anemones, and the like. But in the meantime, this is what's called a soft coral. And as with many of the corals that we've been seeing, it's an octocoral. It has 8 feeding arms. It has this it's a colonial animal, has this very unusual shape.
Come inside if you have any more video.
That'll help, Tina. She actually notices the size of those. I've forgotten. I always get mixed up with my zooids on these things, with the little bumps in between the tentacles. That's some of the things that she looks at when she's trying to ID these.
But you can see that there are projections, the sort of what's called pinnate tentacles that come off of the feeding arms. Often the whole thing is just referred to as pinnate feeding arms.
Yeah.
And the mouth on each one of those, you can see the little connection from the mouth all the way down the stalk of each of these polyps, and they combine into the body there. So it's shaped somewhat like a giant orange mushroom.
So Scott is for me. Those little bumps are siphonozoids. I always mix them up. So that's what they're called. They're actually, if I'm not mistaken, they're kind of a modified type of polyp, but that may be incorrect. All right, so Tina's in the event log, but she's not giving us any help. Come on, Tina, you can do it. I know you can, because she could be logged in and she may be doing something differently and not watching exactly right at the moment. Scott says the siphonozoids are one type and the autozoids are the other type, which I assume are the bigger things. So thank you, Scott.
Yeah, let's frame up the organism.
Okay, and he's providing more information. The autozoids are the typical feeding polyps. Siphonozoids are reduced polyps that lack tentacles. And that's clearly what we were seeing. Very reduced polyps, so we're liking the tentacles versus these beautiful ones coming off of it.
I've been seeing these for over, I think, 15, 20 years, and every time I see a new one, it's beautiful. It never gets old.
All right, Tina, you can have some fun. I'm going to go ahead and ID it, and then you can have fun correcting me. I think it's Pseudanthemastus. That's my best guess. But we will wait and see what you have to say. A lot of times it's whether this is Pseudanthemastus or Anthomastus. Pseudanthemastus, of course, the name is sort of referring to false Anthomastus versus true ones. That's a type of soft mushroom coral.
So I have to head off for an event. I look forward to speaking with all of you very shortly.
Yeah, no, I'm wondering whether I got that right. Could be Pseudanthemastus, or could be Anthomastus. Could we take a look at this coral, Pilot?
Absolutely. Coming on in.
Yeah, this looks new.
Okay, hold that.
Is this a primnoid, maybe? We're going to wait for a moment. A closer view. This is new for the dive. Yes, this is a primnoid. It's in the family Primnoidae. It's another type of octocoral. I think this is the first one we've seen. I think there's a mysid that's hanging on to it, that little red crustacean-like thing. Now, which genus of primnoid? I'm not... Boy. Yeah, it's possibly Narella. Scott concurs it's definitely a primnoid. It's hard to go a little bit more further than that. And I think it could possibly be a Narella.
Thank you, Pritch.
So Scott thinks Narella is the safest guess on this particular one, and thank you very much, Pilot. I think we're good with that.
Come on up, video.
He's noticed some of the characters of the genus, that the polyps are generally oriented downward, and that is they kind of droop down toward the base a little bit. And scales are large and overlapping with, and then Sako Matsumoto also commented it as three polyps per whorl. So I think we're certainly in agreement on it being a primnoid, and I think we're in reasonable agreement it was perhaps in the genus Narella.
Just do a snap here.
Yeah, looks like it's a brittle star.
Yeah, come on out, video.
Up and over, Josh.
Up and over. can't hear you, Chris. I'm not sure what.
We switched headsets. They've got themselves need to get reorganized back there.
Yeah, pilot, I wonder if we could take a quick look at that sort of sponge there on the rock right below the lasers now. Yeah.
Come on in video.
There's a little coral next to it too. Yeah, this is another one of these things that I was a little shaky about. It's a demasponge though, for sure. They could be Stellodorix. Yeah, thank you, pilot. That's good.
Come on now.
You could sort of see how the rock here looks very, very consolidated, almost like it's a sheet. But I think what's happened is there were pillow basalt formations that are when the seamount was being created originally, and it's so old, probably on the order of 70 to 80 million years old, that a lot of manganese crust is kind of glued in all in around the pillow lava formations, making it look like it's one single surface when it's probably more.
Get the opioid.
Yeah, let's go ahead
and
take a look at the Ophi. This is a brittle star.
Oh, it's brittle.
Good.
Just making a break for it, Josh. Gotta cut him off there.
Surprising how quickly they can move.
Especially as cold as it is.
Come on, Chris, go for it. Tell me what he is. He's getting, Chris, next, we switched seats and he's getting ready for a live interaction, but I need help with these guys.
So last expedition, Dave?
He's going to, he's going to write down an ID right now.
Start eating a small baby squid.
I saw that. That was wild.
All right, he believes it's in the family Ophiacanthidae. So thank you very much, Chris. He's definitely multitasking at the moment. I'll try not to bother him too much.
You see those machines that have themselves teach themselves how to walk?
Yeah.
That's what these remind me of.
Probably get rid of lasers if you're gonna follow. There you go.
Let's come wide.
Yeah, and how they don't seem to be very good at it, and yet it works.
Yeah.
Should thinking about another move for pacing wise?
I think so. I don't think you and I can do anything Josh can't keep up with.
Another one of the bolosmo sponges that we've been seeing today.
Although he's got the manip in his lap, he is ready to
go.
Snatch.
He's always ready.
Snatch and run. He's poised.
And I wonder. I was wondering if that thing that's to the lower left that just went off screen, kind of reddish. I wonder if we could take a look at that.
Sure.
Could be a black coral,
please.
actually.
Range 30 meters, bearing 000 degrees, speed 0.2 knots.
Yes, it is.
Black sponge, black coral.
Yeah. So this is an Antipatherian, a black coral. And again, I'm going to call on maybe Scott or Tina to help with the ID. I'm going to look in my guide as well that they've helped us develop. Let's see if I can find a match for this one. Okay, I have a candidate. Scott thinks it's Heteropathes, and that's exactly what photo I've got up on my guide right now. I think Tina might have been going to Heteropathes CF or close to Pacifica. So thank you very much, Scott.
Thank you, Bridge.
All right, let's frame it up. Let's come out video and frame it up.
Go ahead. Yes, 135 degrees looks good.
Couple dead sponge stalks. A little brittle star on the top of that one, kind of similar to what we just imaged a few minutes ago.
Let's get this guy, snap in.
Oh, we've got a squat lobster and a different one. Good eye there, pilot. So this is, I don't know which one this is. I got to refer to the Okeanos Animal Guide again here to try to refresh my memory on what the name is. Tentative name. I think this is an undescribed species of squat lobster, and Mary Wixting down in Texas A&M has been helping us a lot with the arthropods. She and her colleagues.
You can come in any tighter. Go for it.
The way this works often is that a lot of these images get passed around. So somebody's watching, gets one of the images and sends it around to all their colleagues, and they all kind of weigh in on it and get a consensus of what they think it is. It's a really valuable process for us, obviously. I think this is what we're calling Galathioidea long-armed spiny, which means that, I'll translate that for you, it's nobody has a clue. So we'll leave it at that.
Alright, we need a good video. Alright, let's come full wide. Swimmer. Let's get the swimmer if we can.
It's a speedy bugger. Look at him, boss. He's still there.
Just gonna get some of my prop wash, I think, unfortunately. No, I don't know where he went. He's gone.
So, pilot, you think that was a crustacean? That was sort of my guess. I wasn't sure though.
I don't think it was a squat lobster. I think it was definitely some type of crustacean.
Away from the hexactinellids is that hexactinellids have got microscopic skeletal parts that are referred to as hexactins because their basic structure is a 6-rayed sort of star, if we use star-like structure. The demosponges do not have hexactins. They can have other types of spicules like triactins and monactins, single-rayed things, but they don't have the hexactins, and that's why they're put in a very different group. Just a little bit of trivia for any of you spongophiles out here. Another crinoid that we've imaged before, and this is another dead sponge stock, probably a big bolosoma that died at some point in the past. The ball is eroded, that's at the lower end there, that yellow globe that's usually on some of these. Another Chrysogorgia, I guess similar to what we collected. More toothbrush sponges.
Let's get this guy. Come on in video.
Aha. Okay, so I think it's something called a late megonid. It's a sea cucumber, but now I gotta look this up again because Chris Ma is now quite occupied. Boy, give me 1 second. I recognize it.
Complex gut shape.
Probably, there you are. Yeah, so this is actually not well known. The Dave Posson at the Smithsonian and Andrei Gebruck at the Kirchhoff Institute in Moscow have both suggested this is a possible late mogonid in the family late mogonidae, but they're not really sure. So I was trying to decide whether there's any possibility of collecting. What do you think, Pilot?
We can give it a shot. It's likely to get pretty damaged. These tend to be very soft.
Yeah, it doesn't really. It looks pretty flimsy, transparent.
I mean, I can give it a shot and make an attempt.
If you don't close the jaws,
well, this is one where we can't even go to family on it without a question mark. It might be worth trying for. It certainly fits in that sampling criteria. And I guess we'll never know whether we can collect these things if we don't try.
Minutes. Is that going to be a problem?
He'll have it by then.
Okay.
Or not.
Or not. It will be over.
The tough thing is going to be setting up for this.
Should I get the drawer open first, or are you going to sidle up?
I'm going to come in sideways, set down, and then we'll get the drawer open and
roger that.
Why don't I move us back 10 meters just to stop our motion?
That's wise.
All right, I'll call that in.
So I think I want to grab this before we open the drawer.
Okay.
Bridge, nav. We'd like to move back 10 meters, so that's 10 meters, 180 degrees, 0.2 knots. Good copy.
Might be too low for me to get. Down a little bit. Think I'll be able to reach that, Dave? I don't know.
Yeah, maybe a bit of a stretch. It's almost like maybe you should put your other toe down. Although that's a bit awkward as well.
Thank you, Bridge.
I think that'll do it.
That's doable.
I could try to scoop this.
I think
it'll. That, but it's
I think
on a hard bottom.
It's just going to spin right out of it.
Yeah.
I think an encirclement without completely closing. You know, maybe.
Best bet.
Yeah, play with jaw force a little bit till you get the right feel. Hydraulics are up.
It's always harder to set up than to actually get the sample.
Should be starting to feel that move back south, Dave. All right, so which side do you think, Dave? I think starboard side, maybe? You have both on starboard, or you have port outer, but that the coral and port inner is a little big for the box.
Ready on port, aye.
Or ready on starboard, rather.
Okay, I'll just get them both enabled up here.
Who's set? It's moving. The guy's got it almost.
Give him a little nudge so he floats off and then close around him a little bit.
Oh, he's got it. I can't believe it. I mean, I'm delighted.
Don't say anything yet.
That is amazing. These are notoriously hard to collect.
Okay, the gentle caresses.
Not in the box yet, but
starboard
he got it off the walls.
Port bio, roger that. Port bio extending.
And outer, if you can.
Port bio extending. I don't see much.
Go ahead, Dryla.
Well, you have to extend the pin and then open the draw.
Right. Unknown.
Where is pin? Port bio. Said on. Oh, that's the other valve pack.
You should be on starboard valve pack.
Yep. Starboard.
Extend the starboard pin or the port pin.
I see bio. I don't see pin.
Yep. Bio. So retract and then extend.
Retract.
Extend. And now open the drawer.
Oh,
open
right.
The drawer.
Yes. Yep. Got it. Sorry, Josh.
go.
Nice work.
Fantastic. That is a tremendous job, pilot. Josh, that's just great.
It was a long time to hold my breath.
I was holding it with you.
Dry Lab Nav, that was
oh
port
my
outer.
goodness, that's great.
All right, so retract that pin.
Yeah.
We're not going to open that box anymore.
Right, okay.
All right, you got the waypoint nav?
Yes, I have it.
Port bio is retracted.
What comes after a hat
Starboard
trick?
bio retracted.
Oh. It's not the number, it's the quality.
Something big and white ahead to the left.
Yeah, I'm going to take a look at it.
Great.
I think it's a big sponge.
I think so too. Oh, yep, that's what it is.
Yep, it's another one of these big bolosoma with the yellow bases. No idea why it's got a yellow base on it and why this one variety has got yellow tones in the base. Sometimes you can get the ball on top to have a yellowish tint as well. Usually it's at the stem, which is really beautiful.
We once saw one of those as big as Little Hercules in Indonesia.
Something that we call Bolosoma new species 2. Again, for spongophiles out there, like me.
Okay, I'm getting straightened out here. Brought my head around north.
Okay, want to get going north again?
Chrysogorgia that we just passed by. Another one, another beautiful glass sponge.
Bridge nav.
Giant sponges.
Giant sponges, giant yellow lollipops.
Another move, please. Back north, 30 meters, bearing 000 degrees, speed 0.2 knots. Good copy.
I had forgotten about the pin engage, disengage routine with the bio box.
Yep.
While you were going hypoxic there.
It's all right.
It looks like you got him without injury. I
hope so.
Possibly.
I was trying to avoid the blades.
Yeah.
So.
Well, he's pretty squishy, so you might have.
But they're very, like, if you, Dan grabbed one, a bigger one, and he, little slice with the blades, and then the way up it just
exploded.
Exploded.
Thank you, Bridge.
You see some more of these beautiful stock sponges in the background there. Again, the bolosomas. Probably at least another 4 of them on the slope. More of the chrysogorgids really corals. These are the ones that are sort of in the center, upper center.
Lobster we grabbed.
Very fine.
Yes.
A couple years ago.
Group of octocorals.
Yeah, that's right. You had to move so stealthily that he didn't jump off.
And then he jumped off. And I made a mistake and I dropped him.
Right.
And then I picked up the piece of coral.
And coaxed him back on.
So again, if you're just joining us, we're broadcasting to you live from the NOAA ship Okeanos Explorer. We're on an unnamed seamount, probably about 120 miles or so north of Johnston Atoll. We are currently at 2,488 meters depth. And we are surveying the flank of the seamount, sort of a ridge, possibly what they call a rift zone ridge that's extending out from the summit of the seamount along its flank.
Just 1 minute to check math.
Just cruising up along this ridge and trying to document the animal populations and the topography and geology we encounter. Nobody has ever been on the seamount before. This is the first time it's ever been explored, and you're seeing it at the same time that we are. I'm very happy that you could join us here for what's proved to be a really interesting dive. We've got we got 2 biologic samples, one of these corals and that sea cucumber that we just collected, and we've got 2 rocks already. And we're just continuing upslope, seeing what else the seamount has to offer.
Yeah, I'm satisfied. Brief moment of panic.
What's our bottom time?
15:20 will leave the bottom, and
15:20 local.
Local, yes. So about 2 hours left.
What are you trying to average? 0.1?
0.08 will get us to our final. Let me target. Oh,
These are chrysogorgid corals in front of us as well. We've been seeing these all day.
325 meters from the waypoint.
One variety that's sort of columnar that's in the lower right, and then there's one that's on a sort of a stalk that's going off in the upper right as well.
A little less than that. But
this is a very diverse family. It's also one that's being actively worked on to try to improve its taxonomy. And hopefully in a short period of time we'll see some significant significant number of new species in the family. It's already pretty speciose, but I think the Okeanos has collected a whole bunch more new ones over the course of 3 years working here in the Pacific.
All right, we've got Dave Casagrande piloting now.
We're pausing for a moment. We had a bunch of musical chairs in the front row here. And I think Josh and Dave had switched places. So Josh is now navigating. Dave is on the pilot stick. And the other Dave is co-pilot. So we have Dave and Dave piloting here today.
Uncle Dave.
And in the back we have Chris and Chris providing the science. And next to us in the nav chair on the far left is yet another Chris. So, yep.
Would that be a triumvirate?
And then we have
Chris's?
Tara, not Chris to my right. So we only have 3 Chris's. But, oh well. We can't all be Chris's. I know you're a little sad about that, but... So that's right. I can be your honorary uncle. That's right.
Somebody's tugging on me.
That's true.
Somebody puppied off.
I'm trying to get back.
Okay.
Is it? So, just pointing out that one of the we've got where we've got all types of Kellys, Daves, Chris's, everything on this ship, this cruise. So it's
centering up on you.
Going to be only one rolling. That's correct.
All right, gentlemen, I'm going to put in another move
That sounds
So
great.
what's happening right now and why you haven't heard Chris Ma's voice for a while is he's standing by to do a live interaction. I believe this is with IRC. They're having a science camp at the Anoye Regional Center.
Like to repeat the
Ford
previous
Island
move, 30 meters at a bearing,
a little bit of
000 at a speed,
so Roland and Kelly Elliott and Chris Ma are all standing by ready to do a live broadcast here, and that's why you're not hearing Chris Ma.
That's correct.
Video, let's
talk about things or even get wildly excited about that sea cucumber collection. But he was sitting next to me, and I could tell that he was extremely excited about that.
Okay.
Yeah, so
even though you didn't hear it, he was.
Video.
And I have a feeling that he won't let me come near it when it comes up. He's going to handle that baby by himself.
Sure.
Yeah. On the contrary, Chris.
Thank you, Bridge.
I'll come down.
Those things usually are really
to wander
slimy and messy. You'll be able to work on those with every little bit of effort on your own. Thank you very much.
Yeah, the manip cams are really good for giving you a sense
Pilot
of your
Dave, could we
altitude.
take a look at that branching coral that's on the rock, that little white one, if we get a chance?
Top center?
Yeah, top center, that's it.
Sure, we can do that.
Maybe something different.
Also have one of those nice spirals in the bottom left. Okay, video, you can come in on this.
Okay, that's a really good view. So this is going to be a coral in the family Primnoidae. It's an octocoral. And I'm pretty sure we can go to genus on this one. I'm very certain it's Narella, and it could actually be something that we call Narella macrocalyx. But a Narella, I think, is very comfortable. The polyps are all kind of drooping down toward the base, and that's sort of one of the characteristics of Narella that helps identify it. And also, believe it or not, the number of sort of ridges or body scales on each of the polyps. Narella tends to have 3 or more, and I think we can see sort of 3 as well along these particular polyps. So I'm going to see if anyone from the event log can weigh in on this as well, but I think it's a reasonable idea.
To your port is another sea cucumber.
Okay.
You look in your port rail cam, you can see it.
Yes.
I think it's the same. Might be the same one we just saw.
That's good for Science Pilot whenever video's finished.
Okay, video, whenever you're done. And there's a sea cucumber down here that we're going to try to get a quick look at.
Oh, there's another sea cucumber. I wonder if it's the same one. It looks a little different. Maybe we can do at least a snap zoom on it.
Come in quick.
And
doesn't have the
he does not have
spines.
the long projections coming off of his back, I don't believe. So superficially, I'd say this is something different. Would you shake your head one way or the other, Chris, so you concur? And he throws up his hands and basically goes, I don't know. So he's no help. I believe this is a different one. It could be in that same family, Late Megonidae. I think what characterizes these are very small, numerous tube feet, and you can kind of see a lot of tiny tube feet underneath it there that it uses to walk around on. But I'm going to take a quick look in the Okeanos guide. I think that's characteristic. I think this is what Dave Pawson at the Smithsonian told me about this family. He's one of the foremost experts in the world on this, on sea cucumbers. And I was able to visit him a few months ago. He's in there. So I think we're going to go ahead and call it possible late mogonid. So thank you very much, pilot.
Great video.
But a different one than the one we collected.
Are these sponges getting larger? This one's pretty impressive.
I think everything is getting bigger. I'm now seeing my event log was frozen for a little while, and now I see some comments here. And Les Watling and Asako Matsumoto also are fairly confident that Primnoid was an Arella, so that's great. So you got three of us all on the same page. Thank you very much for weighing in on that. You're looking at another one of these beautiful big stock sponges, glass sponges. You've been seeing these all day.
Thank you, Bridge.
There's another Chrysogorgia, a couple Chrysogorgia octocorals. We've gotten some good imagery of these today already. There's a bamboo coral in the back. It looks like one of the ones we've also got some close-ups of. It could be another Primnoid next to a Chrysogorgia. It might be worth a shot zooming in on this cluster, pilot,
okay,
right here on the side of the rock. Looks like we got three different things going on.
Attention all hands. There will be a ship store meeting held in the forward lounge in 5 minutes. Again, that's a ship store meeting held in the forward lounge in 5 minutes.
Who would attend such a thing?
So I think we've got two Chrysogorgids that are bracketing a Primnoid. And that Primnoid could be another Neurella. It almost looks like Neurella musicae, but I'm not positive.
Maybe on the center one.
Anyway, the Chrysogorgids, I think we could get a close-up of all of these because they're all pretty interesting.
Okay.
Why I sort of went to musicae is it looks like a lot of the branches are coming out very close to the base, even though there's some sub-branching going on there, but it looks a little different to me, so you gotta back off just to Neurella. You've also got some anemones and brittle stars that are clinging onto the branches, possibly an Ophicanthidae, the brittle star. I'm going to also wait and see if we get some help on this from shore. But I think it's an Neurella. Let's see what others have to say.
Video is that full?
Okay, and then how about the one to the lower below? And this looks like a planar Chrysogorgid, if I'm not mistaken. So Les Watling and Scott France are still on. I think Les is. Scott may have finally gone to bed or something because it's pretty late for him. But Les, can you give us a hand with this? This looks planar to me.
Any particular part of this watch lead?
There's
a lot going on.
I think this is a pretty good view. I think I'm not sure exactly what he'd be looking at, but you've got branch points here and a bunch of things. I think the Tim Shank would probably appreciate if you slid over to the left a little bit and took a look at that brittle star, because Tim Shank at Woods Hole is quite interested in the associates for corals. Also looks like there's a little squat lobster in there.
You can come in, video.
Asako Matsumoto has already commented on this Chrysogorgia, saying it's very unique, and I would agree with that as well.
And I would also add Chris Ritter.
Okay, Les says, does not branch in a typical planar way, which is more dichotomous. So that's pretty interesting. Yeah, that's good for science. Thank you, pilot.
Okay. There was one more, but I think we've seen a lot of that
I think we have two. I agree.
Great.
So we could probably push on. This is possibly that Chrysogorgia averta.
Right over the top of you.
Yeah.
At least it's a species of Chrysogorgia.
Once you start pushing ahead, I'm going to call in another
Good.
30. Bridge, RVNAV. We'd like to repeat our last move, 30 meters, bearing 000, speed 0.2 knots.
Let's see what this swimmer
Yeah,
is.
we've got a little something here in the water, don't we? Hanging
Come in,
up.
video. Try
That's
to chase
correct.
this
Thank
a
you,
little bit.
Bridge.
He's distracted. You to speak up.
Video, let's try to see what this is in the water column. It's starting to lose it.
Just go up with it.
There's some type of crustacean, possibly. Yeah. I don't know these very well. Maybe some type of a mysid, maybe, I'm not sure.
Thank you, Bridge.
Good
That's
job,
all I
Dave. Stayed with him pretty good there.
Yeah, Liz Watling has concurred that that was probably a mysid, a small crustacean. Pilot, can we get another look at this Chrysogorgia coral? It may be the same as that planar one we saw on the rock a moment ago.
Sure.
Both Lasako and Les were quite interested in this thing.
Come on in, video. Video, zoom on this, please.
I think this is the same thing, but I was going to see if Lesser Asako or somebody else had an opinion on this.
Is it the branching in this we're particularly interested in?
Yeah, they are toward the base too. They had commented that it's showing some dichotomous branching. Yeah, right there is a really good look at it. I think that'll help them out.
Thank you, Chris. So I'm Chris Ma, and I'm a marine biologist from the Smithsonian Institution of the National Museum of Natural History, all the way on the mainland of Washington, D.C.
So, pilot, this might be something that would be worth considering for a collection. I don't know. Do we have anything on starboard side that's open? And we fill both port, right?
We have space on the starboard side, yes.
There are not a ton of animals here that are particularly new, and definitely there's some interest in this one. I think we'd get support if you wanted to set up to collect this one.
Okay.
Somebody just went ahead and called in. Who's that?
I'll get us a little bit closer.
Yeah, Chris, this is Les in Hawaii.
Yeah, hi Les.
I just wanted to say that the pattern of branching in these two flat crescent gorges are quite different. This one is much more typical of what we would expect to see in a planar colony in terms of the way the branching is sort of dichotomous, or you might call pseudo-dichotomous, but essentially each time it branches you get two. And whereas in the other one, it was much more of a central axis, several central axes actually, with branches coming off the sides, and then those branches subdividing.
Pilot, stand by on the collection, please. I'm hearing some comments right now.
And in the first one, also, it looked like the branches could have been a more typical chrysogorgia, coming out at different angles off of the axis, so they weren't all in one plane. But in that particular case, they were all in one plane. And so it kind of is like if you took a typical bush-like chrysogorgia that we normally see and made all the branches come out either to the left or to the right, and not front and back sort of thing. And so it's quite different branching pattern. And the truly planar chrysogorgias that we've seen over the years are more like the close-up that we just saw of the second one, with central axis sort of disappearing and the branching becoming more dichotomous as you go up the colony.
So, Les, would you think the second one, since we've moved away from the first one, is this a collection option, or is it common enough where we should move on and save our collection for something different? Did you catch that question, Les? I can't hear you. I was asking whether or not this would be worth collecting, or is this just something like Chrysogorgia crises or one of these other planar ones, and we ought to skip?
There's a bit of a delay, it seems like. But my guess would be that the first one would have been more interesting to collect than this one. But either one would be good, but I think the first one would have been a better choice.
Yeah, we passed that by. Is it of interest to collect the second one? Because we're sort of standing by to see whether or not this is worth grabbing, or whether we should save that final collection for something else. You still with me, Les? I tell you what, Pilot, let's go ahead and move on. Maybe we'll find another one that's similar to that first one he was talking about. He was more interested in.
Okay, moving on.
Yeah, thank you.
Just a wee bit past you, not much.
Yep.
No panic.
Yeah, sorry, Chris. I think that would be a better decision if we were limited in the number of things we can collect. It would be better to get one that was more like the first one, if that's possible.
Okay, we'll keep our eye out for it, Les. Thank you.
It's a little steep here.
Sako, I didn't see your message, but we'll keep an eye out for these planar ones for you as well.
There might be we already have two rocks.
So, ship stationary right now, waiting for you to push out a little bit.
Okay. Yeah, it's getting a little steep, so why don't I get out ahead first?
By the way, it was the voice of Dr. Les Watling at the University of Hawaii. And he's one of the foremost octocoral experts on the planet, as well as a lot of other things. Does a lot of trawl damage studies and involved in some nodule work and all types of things. He works both the Atlantic and the Pacific, so he's a very very broad, experienced resource for the Okeanos Explorer cruises. So thank you, Liz. Looks
and sunlight only penetrates maybe the top 200 meters of the ocean. For plants to live,
maybe we could take a look at this crinoid pilot if we get a chance.
Okay.
Thank you. Could do a snap zoom. I think we've seen it before, but I'm not sure.
Into the ocean. It's also very, very cold. I believe.
Yeah. I think that'll do it. Thanks whenever video's ready. Thank you.
So, pilot, the terrain has steepened and you should get up quickly.
Okay.
And get a look. And then be ready for whatever is coming.
Oh, pilot, there's a spirally coral to the left. I think it's the first one of these we've seen. We could get a snap zoom on this one too. It's a pretty common one. This is a
I think we saw one more earlier. But,
oh, you did? Oh, then we
can. No, we didn't get a good view of it.
Oh, okay.
Yeah, just. Do I have time for this?
Yeah, you can.
Yeah, just a snap zoom is fine. This is pretty common. The Iridogorgia, possibly Iridogorgia magnispiralis.
Come in. I'm still approaching it.
These are real pretty. Since there's some people watching the video right now too.
Sure. Video, you can come in on this. Sorry, trying to get back to it.
So these, of course, are some of the more gorgeous members of the deep coral fauna down here at these depths. They're really popular just because they're such an elegant animal with these beautiful spiraling branches with their polyps on them all lined up. So thus the name, it's called Iridogorgia magnispiralis, and that's referring to the fact that I think that the spiraling
video, come on out.
Tends to be a little bit larger turns in the spiral. Some of their other species that have much tighter turns in the way they spiral and how the branches come off of them. I'm sure I didn't say that right compared to how Les could probably describe that, but anyway.
Need to sprint off ahead a little bit to deal with.
Got some more Chrysogrogids, but these aren't the ones that were of particular interest. This is Plurigorgia and the Chrysogorgia averta we've seen earlier already today. It's like it's another one of these bamboo Bathagorgia that we've also imaged today. Single whip corals. We are currently at 2,461 meters, and we're still climbing up the crest of this ridge that we're on. Those of you who are not familiar with Okeanos dives, you're seeing two red dots moving across the substrate. These are lasers. We use them for scaling, and they're 10 centimeters apart. So that'll give you an idea of the size of animals if you know that, as the lasers get close to some of these corals and sponges and so forth.
at the Sirius view.
Yeah, I'll go check those out. We've seen plenty already. But
it's like some kind of nice, mature, long toothbrush sponges. We could take a look. At least that's what I think they are. And some big, these yellow bolosoma sponges. There's some more of those showing up.
We want a close-up of the toothbrush sponge?
I don't think so. We've got a couple close-ups of them, and I think we're okay with that.
Okay.
Thank you. Pilot, I guess that's something pretty bushy on the top center there. I wonder if we could zoom on that. This may be one of these planar corals that there was interest in.
Sure, I'll get closer first.
Yeah, thank you. I've been trying to keep my eye out for some more of these.
Yeah, we're okay now. We got up over the
really
nose
sure
of
it's
this
the same
thing,
thing, but could be.
Got time.
Okay.
This may be similar to the second one. Les was saying it has a more typical branching pattern than the first one, but we'll find out when we get a little closer.
Okay, video, come in. I'm still moving, sorry.
Wow. All right, Les and Asako, could use some help with this one. So I'm going to throw out Chrysogorgia crises for this one, but Les and Asako, do you guys have a different opinion? And it may not be crises, but it looks, these kind of thicker branches look a bit like crises. Is that you, Les? I don't think it's him. I think that's good, Pilot. Thank you.
Okay. Video come out.
So you're right on the nose of this thing now. Steep.
Open over. Think we could get moving again?
I'm good for it. You're going to have to entreat the navigator to indulge us with an advancement.
I shall indulge.
Navigator, sir. Would you be so kind?
Bridge, RVNet.
Pilot, could we take a look at what's on the side of that rock right up ahead?
That bridge
looks kind of spiky.
Would it be possible to do
right ahead of us?
Yeah, right ahead of us. It's got a slightly upper center.
Got a bearing 000 at a speed 0.2 knots, please.
I don't know what that is right off the bat. Maybe it's just a crinoid, but
that's correct.
It's looking more maybe a snap zoom because I think it is a crinoid now that you're getting a little closer to it.
All right, video, just come in.
And it could be one of Chris's creatures, Chris Moss. And it is. It's a Brazilian sea star. It's one of Chris's sea stars, finally. Yeah.
I'll try to come back to it. Just come out for now.
Thank you. Move initiated.
Come back in.
You can get even tighter after video gets their whole colony shot. That would be great.
Okay. Video, do you have any more?
Yeah, so I'm going to take a wild stab at this because Chris is engaged in the live interaction. I think this is in the family Freyelidae. It's in the order Bracyngidae, but I think it's and possibly Freyastera, I guess, but I'll check on that. But it's a deep water specialist. And can hold it for a second longer. I think Chris is going to talk about it to the line of interaction.
Okay.
Squid with long arms. Is that right? Well, on this expedition, I don't believe we've seen any squid yet, but certainly.
Yes.
There are many species of squid that live in the deep sea, and usually what happens is you have
I think that's good, pilot. I've got an ID on it, a tentative ID.
Okay, video, you can come out.
Thank you.
There are, of course, lots of different types of squids, and so many of them might have really long arms relative to other types of squid that have short arms. So it really does depend. But there are a lot of squid that have very bizarre appearances in the deep sea. Some of them can actually project.
I believe this is our largest of these sponges yet.
That's a whopper. So this is another one of these stocked bolosoma glass sponges, but you can see the lasers right underneath it. The pilar's putting it right underneath it. And you see how big this thing is. This has got to be a...
That base is 10 centimeters.
Yeah, the base is 10 centimeters alone. It's probably at least a meter high or so, and the ball itself is probably at least half a meter across. That's a big animal. And yes, it is an animal. It was a question we had the other day, so...
Nav, are we moving?
I thought that was live.
Nav copies. Thank you, Bridge.
Might as well take a look at this unbranched coral. I think it's another bamboo, but we could take a peek at it and just confirm that.
Sure.
While we're waiting here for the ship and the cereals to move.
Video snap zoom, please.
Yeah, it does. Yep, it's a bamboo.
Hey Dan, just an
that'll
FYI.
give Les
we have a light squall going
chance
over
to
the
look
ship
at
right
this
now.
and perhaps come up with a better idea.
Good to know. Okay.
Do you want to get any better views of this?
Excuse me?
Do you want to get any better views of this one?
I think we're pretty good, thank
Okay.
you. Here's some
Come on out, video.
Is the ship moving?
I believe it's just stopped or about to stop.
Okay.
But you should still be swinging. Thank you, Bridge.
Yeah, Chris, this is us. I couldn't get quite a good enough look at that bamboo, but it seems like well, here comes another one. It seems like there's like this huge population of this one particular type that we would put into the C1 clay. Most of them are pretty small and young. This one looks like it's starting to curl a little bit at the top, and suggesting that this is a much older animal. So it's hard to say. We don't obviously know how old, but I think that this is a much more mature specimen than the others that we've been looking at.
This is, so are you in the ECC, Les, or do you have a delay right now? Because we're looking at a very tall bamboo at the moment. Is this the one you're talking about?
That's correct, thank you.
Yeah, that's right. This really tall one. Yeah, I think that in my lab here, I'm getting a delay of about 6 or 7 seconds, even on the low latency picture. But yeah, this one looks pretty typical for this group. C1 clay starts to curl at the top when they get older.
Great. Thank you so much, Les, for calling in. I'm actually logging your ID right now in C-Scribe.
Okay, video.
Les, I also don't know if you noticed, I think I saw some eggs in that previous one we saw at the base of the polyps near the top. You probably noticed that because you usually spot them. But it's possible that that particular colony might be in a reproductive state or starting to come into a reproductive state.
Yeah, I think that reproduction in these octocorals seems to start pretty early. In other words, when the colonies aren't all that tall, and we don't know really, how old they are when they start to reproduce. But you're right. I mean, there were clearly eggs in that one, and in several of the other ones that we looked at had close-up views of. You could see the eggs at the base of the polyp. It's possible that they reproduce pretty continuously for their whole lifetime once they get started. It's possible that when the colony is like 20 or 30 centimeters high, that some of the polyps will become reproductively mature, and then they will go through multiple cycles of egg production. We actually don't really know much about that because obviously we can't come back to the same colonies every few years and sample. But we do see reproductive products in the corals over a fair long period of time and over the whole of the size range that we usually encounter.
Yeah, and Les, do you know about how many eggs you typically see in these polyps? Because you could almost get an estimate at least of batch fecundity by essentially just counting the number of polyps on one of these whips, and then if there were, say, 5 eggs per polyp or 10 eggs per polyp, then you could kind of get an idea how many they're putting out at any one time. But is that known? I mean, has anyone looked at that?
Yeah, there's been a little bit. I had a student at the University of Maine a number of years ago who started to look into that. One of the things we noticed was that in any individual polyp, the eggs would be at different developmental stages. And so you might have like somewhere between 5 and 10 eggs in a typical bamboo polyp. But some of the eggs would be just beginning to develop, and others would be looking like they were getting close to maturity, and you have everything in between. So it's possible that they produce eggs on a really continuous basis. And obviously we don't know anything about spawning, how males, we do know that colonies are either males or females, but we don't know how reproduction and fertilization actually occurs, because that's never been observed in the deep. There have been some observations off of Newfoundland in Canada and the Atlantic that suggest that some of the corals are capable of having fertilization on the polyp and then brood the young, hold the young, like hold the larva basically on the polyp until it's more mature and ready to swim away and then settle somewhere.
Do you want me to stow your lower swing arm? Or?
Yeah, well, thank you very much, Les. I really appreciate that. For those of you watching, we now got a fish on screen, so I'll go ahead and describe this. This is a rat tail in the family Macruridae, and it looks like it's one that we've seen before that we're tentatively identifying as in the genus Kumba, but that's not absolutely certain. There's a couple other genera that are possible candidates for this fish, but that's beautiful imagery, and I'm waiting to hear if some of the fish people like Tracy Sutton, who's on, or Ken Sulak are going to weigh in on this fish. Again, they sometimes have a little bit of a delay, but I think they're probably looking right now.
Misbehaving video, you want to try to come in tight?
Nice thing about this one is if it is one of these Kumbas, normally they're just zipping around and they're not posing very well, and so this one is definitely a poser. Hello, do we have, is that Les signing off or maybe somebody signing on? But anyway, I'm going to look in the event log and see if we can come up with any other comments regarding this fish.
How much left in that move? Just curious.
About 3 meters, maybe.
Okay. And another.
Okay, I don't see a lot of dispute for my ID so far. So I guess it'll stand for the moment until we can hear a little bit more from people after the chance to really examine
Starting to look down at you, Dave.
Okay.
The imagery. Thank you very much, pilot and video. That was great.
Not hitting that arm. Great.
Yeah. And it doesn't make sense in making for half an hour. Yeah. But that was the.
Thank you, Bridge.
So Tracy's deferring to Ken, and
so why don't you go ahead and push it up slope a little bit?
Sure.
Ken says head shape looks like Kumba, not Nazumia, but I do not know the Pacific genera that well.
Just behind
Thanks
Sirius,
so much. This is
another
Ken
half turn in
Sulak,
the opposite direction.
Who's one of the foremost deep sea fish experts on the planet, and he's been regularly helping us with identifications.
So is that suggesting it's like a set in the
So
tether?
I really appreciate that.
Maybe
Thank you,
somewhere
Ken.
in the middle here.
And Tracy,
Get
both
laid
of
on
you.
deck like that, but, well, it wasn't on
I
deck.
think we need bigger.
Yeah, why don't you line up with Sirius here and push over to your port side?
Sorry.
You need what? Josh?
Bigger crates.
Yeah.
Or a stretcher mount.
You know, that's the The one complaint I hear about those tether boxes, they're just not big or heavy enough.
They're one of these couple chrysogorgids here, like 3 or 4 of them there. These we've been seeing this type of coral
west side
on
of
this
this
feature
ridge.
for most of the dive.
I don't see much of any current, so I don't know if that's...
I'm just saying, like the sponges and all the, the corals we're seeing on the west side.
I'm saying that, thinking that would have made sense if we saw current coming over from that direction, but I haven't seen that.
Got a couple dead sponge stalks, and there's a couple of these toothbrush sponges as well going across the bottom.
Gotten a little high again, but...
We're now at 2,433 meters down along this ridge. That's stalked white sponge is a Calophacus. We have seen that already today. We've imaged that one. We're of course keeping our eye out for some new things that we haven't imaged already on this dive.
Now, how are we doing pacing-wise?
How are we doing pacing-wise?
Yeah.
We have 230 meters to go, and about an hour and a half.
It's not bad.
It's not bad at all.
Yeah, that's not bad at all. Thank you very much, Josh, for that update. We're doing well.
So when you're comfortable, I'll put another move in.
This is Chris Ma, and I'm back on. Thanks for being patient with me.
I think we could do
Putting
that.
up with my very loud voice.
So, got something over here on the right, pilot. If you see that,
5-armed crinoids.
I'd like to repeat our last move.
Big one.
Yeah,
range
we saw one of those
30
earlier.
meters, bearing 000,
Interestingly, one of them seems
speed
to, one of those arms
0.2
looks
knots.
like it's damaged or something.
So this would be the Pentametric Crinus, is that right? The 5-armed ones? It's the first one I've seen.
Yeah, I know it's.
That's correct. Thank you.
It's got that distinctive stripe along the arm.
I'm gonna try to come in for a better view of that.
Without my
But I do know
Okeanos animal guide images as we speak. And you too can see these images if you go on the Okeanos website, because there is an awesome animal guide that's going to be updated shortly with even more photos. There's already 1,700 photos on that website.
Video come in.
Our next version is going to have 2,900 images. So.
But I did mention earlier, I think, that it was unusual for crinoids to have only 5 arms.
Pretty unusual for them to have 4 arms, too, isn't it?
Looks like one of them has been
yeah, one of them was damaged, of course.
You saw a fish earlier, too, right? Sometimes
yeah,
fish will bite
lasers
crinoids.
That's pretty, well, okay, that's only fair, because your sea stars bite corals, so what the heck?
I think you're zoomed past them already.
It might be, yep.
That may have switched them off. Nope, there you go.
No, my best, I'm going to go with the Pentametric Crinus for now, at least with a question mark in the log.
Let's get a close-up of the center.
As Chris pointed out, it had, the one he saw earlier, which I missed, had 5 arms.
1, 2, 3. Yeah, see, I think that's a stump right there. It's probably lost one from something biting it off. So this is what we call sublethal predation. Something bites off a chunk of an echinoderm, and echinoderms are by and large known for their interesting abilities to regenerate. Yes, that looks like it, Pentametrocrinus. This is one of the more unusual echinoderm, or crinoids, one of the more unusual feather stars, because, like I said, it has 5 rays instead of the many, many more that most other crinoids have.
So, Chris, we did get one of those sea stars. It's actually showing over there on the Clipper screen, Tara screen. I went to Fraility, but I am not sure. They zoomed in on it, and I didn't see the ridging on the arms. That's why I thought it could be a Fraility.
Come out video?
Yeah, that sounds like it might be. Looks like it might be right. I'll take a look at the arms more closely later. But Frailids are often Fraila and other Fraila type Brisinghids are often found in much deeper waters. But it could be Hymenodiscus or some of the other Brisingid types that sometimes the you see some of the spaces between the arms that acts as a diagnostic character, which is indicative of a skeletal feature that you need to look at on individuals. So it gets complicated very quick with Brisinghids, so I'll have to take a look at it.
So Nav, if you have a minute, if you look back about 2 pages on the left side of that notebook, I have some notes on importing sound speed profiles into TrackLink.
Pilot, could we take a look at that white sponge-like thing to the upper left corner?
Sure. I'll have to be a little more specific as I get closer.
Yeah, it's this, not the bright white, the kind of milky white stock.
Okay.
Yeah,
stock thing.
I looked at it the other night. Seems pretty straightforward.
Great.
Question right now is how many data points to provide.
Well, enough to accurately describe the curve and no more.
Well, of course, but how many is
this
that?
Sort of column with half a palm
I
out.
can do a 1
right,
second.
so this is going to be in the family Pheronematidae. It's not in the genus Semperella. Those are the ones that are out on the sediment and the manganese nodule field. But it's related. It's in the same family, I think. And this is one that at some point we really need to collect. We don't have to do it today if the folks would not want to keep their eye open for that planar chrysogorgid, for example. But I'm not sure what sponge this is except for the family. It could be Polyopogon, but I'm not positive. And I'm only mildly comfortable that it's even in the family Pheronematidae. I think it is. Looks like it's sort of bent over, so there's a scooped side on the opposite side that we're not seeing, and we're looking at sort of the concave surface of this one. I think we can probably let this one go, but it's interesting. It's an interesting sponge.
Okay.
And thank you very much. I appreciate it.
So rays in a piecewise linear sound speed profile follow arcs, and if you get beyond linear, they get messy.
Think a bridge.
So basically, it's just a refraction related to the
there's
density
a dead
gradient.
sponge stock with some barnacles on it. It looks like.
Oh, that's neat.
Yeah. Barnacles have found a nice place to perch.
You actually get profiles, Dave, where the sound will bend so much that it bounces right back up off the surface.
There's another one of the Brisingid sea stars, I think,
angle.
isn't it?
Looks like a crinoid to me.
A crinoid?
It's important for submarines.
Yeah. Do we want close-ups on any of this, or keep moving?
Could take a look at the crinoid.
Sure.
Now, for us, Josh, we're dealing with primarily a direct
would
ray,
be fine,
though,
I
so
think.
the effect is minimal.
It's a sea star. I think it's a sea star.
It's a crinoid.
Okay.
Coming on the.
You're on. I'm not betting you a dinner this time. Oh, doggone it! You're right. Darn it! There's no sweeter victory to actually get one on the Echinoderm expert, but I have yet to succeed. So, oh well, I'll keep trying.
Not to worry, Chris. Next time I'll actually challenge you. You can buy me another pork chop dinner.
Yeah, you noticed I wasn't gonna say anything about dinner this time.
All right. Let's come out.
By the way, the crinoid feather stars are in that other. I finally spotted them in the annotation. Oh, you got it. Great, thank you.
Set her up again.
That's an interesting sponge stock with some manganese crusting on it. Off to the left there.
Is that what that is?
Yeah, I think so. Yeah.
Let's go look at that then.
Not that interesting. It's a dead sponge stock.
Well, it's got manganese on it.
Yeah, I guess so.
You mean the front row's not the only one that's yawning?
Let's get a quick zoom on this and see if it is in fact.
Let's poll the audience. Who thinks this dead sponge stock is really interesting?
Come out a little bit. I'm not stable.
I'm in the process of passing you by, my brother.
Okay. I guess we'll just zoom out and move on.
Thank you, pilots.
So Don Kobayashi at the Inouye Regional Center is with Noah Fisher. He says, join the event log. Hello, Don. Glad to see you.
Let's check out that crinoid up at the top there. It's a different one.
Yeah, that is
might
different.
as well.
Just blast. Fed the fishes is the technical term for that.
What was it?
I just fed the fishes.
Oh.
The ship is stationary.
Oh, okay.
We're just swinging at this point.
All right. It's just whenever I see
this is interesting. Look,
mountain coming at the top of the screen.
What is this going to be?
I don't know.
It's disconcerting.
But it's interesting
come in a little bit on this.
to note that there are other crinoids along the way there. Oh, interesting. And look at that.
So what's really cool is he just whooped me on the sea lily or unstocked crinoid, which I thought could be a sea star, and now we're looking at a stocked crinoid, which is called a sea lily, right?
Yes. So this is a different morphological form of the same group of animals, the crinoids. And so these actually sort of are an older form. They have a stalk, and some of them can move, but most of them are anchored. This one's really interesting because the stalks, which are usually clean, actually have animals that are living on them, and in this case, it looks like they're hydroids.
Stalk.
So, wow, yeah, it's just infested with them. As a general rule, stocked crinoids live in the deep sea, and they're rarely encountered. They don't occur in shallow water, or if they do, they're at the deeper end of shallow water. And so a lot of my colleagues who study them have very few specimens to work with. And I'm told that often it's not unusual for them, for every new specimen that they encounter, to find a new species. Have we collected our quota today?
We've got 2. We can go as many as 3. Shall we pull the event log? What do you folks think about this as a possibility? We have about an hour left in the dive.
Bridge, RV nav.
Yeah, let's check to see whether it's even possible.
I'd like to request a ship move.
I think there's going to be a ship move
Do
here.
15 meters.
If not, that's fine. We can get another one.
No, actually,
delay that. Stand by.
We were considering moving back in case you were interested in this.
I see. So I don't see any of the filaments on the arm, so it's probably not Naumacrocrinus. So then Bathycrinus.
Let's also get a shot of the base.
Ship move at the moment, Bridge.
Yeah, let's...
I'm sorry, you wanted the base?
Oh, yes, I'm sorry. Please, if you could take a zoom on the base, that's also something that's important for people who study these.
So is this what we think we're looking at?
Yeah, copy that. You're clear to rotate.
I don't know. It certainly got a whole lot of stuff growing on the...
Yeah, we've got, even these things.
Okay, so it might be...
I think you got to move, Dave.
Yeah. Well, no,
the ship's got to change its heading.
This is shallow, and it's got the filaments on the...
Come on out, video. We need to move.
Yeah. Just getting a little close for Sirius here.
Yeah, I'm looking
why don't we
way
skip this
back.
Okay, let's we can skip this for now, and then we'll come back for it.
Head
I
up
think
the
we have some
hill,
good imagery,
bring my head
and
back north.
we can negotiate what.
Just shoot
what we've
straight
seen
up the
before.
hill.
Okay, I was going to center up as well, but I'll just go straight up.
Centering up on me is I'm looking over my shoulder at you, so just get up the hill.
Yeah, so if you lateral to port as you're going up, you'll
doing
center
a little
up.
bit of that.
That away is what you're looking for.
All right, that looks better.
Well, till you look at my picture.
Oh,
so just to give you an idea, you're going to hear a little more chatter in the background from us, I think, in the back row here, because we're not only are those of you on shore annotating in C-Scribe, but we're also trying to help in real time on the ship to try to get a really complete set of annotations for everybody's reference. And we're getting used to and adjusted to what's required to do that, and amongst the other types of things that we're doing here as well. And Nicolette next to me on the left is really helping out as well as you are on shore, because she's just showing me all the people that are annotating in there, so we really appreciate it.
Thank you, Bridge.
Okay, well, actually, we're straightened out here, and we can
how about
resume
that,
looking for things.
Yeah, how about that mounding sponge that's to the upper right, which I think is going to be a real solid member of the Poliopogon genus of glass sponges. This is one of these, again, another weird one. We could just do a snap zoom on it just to confirm that's what it is.
You know, the ROV nav is way off.
Yeah, and so strangely enough, we call this one sometimes Poliopogon holy. Like, can you sort of follow why we might call it Poliopogon holy?
You notice this happens.
Mainly because of all the big holes in it?
You've got it. There it is.
You notice this happens when we're close to the edge of a cliff.
So just like you, we're identifying things, based on southern morphology,
we're on
right?
like a ridge.
You really can't escape that conclusion with some of these.
Yeah. And I'm thinking it's like bouncing off of hitting the ridge and then bouncing off something else and coming back.
Yeah. They call that multipath.
The intricate structures on the inside of that's a glass sponge.
But
right?
I mean, Sirius is pretty good.
The intricate structures on the inside of this glass sponge are really remarkable.
All over the place.
Is it every ping that's bad?
This is a real weird one,
yeah,
too.
I mean, it's jumping all over
very
the place.
Unusual,
D2 is like way off.
It's very delicate for that group of sponges. But I'm pretty sure that's what it is, because those are the ones that produce these kind of massive mounding type of sponges down here.
What does the name? So poly means many. Does Pogon mean?
Serious.
You know? Does it mean whole?
No, because there's. I don't think so.
Pogo. I'll look that up.
I've got my Latin root thing with me in the room. We'll take a look at that. So thanks for that question, Chris, which I couldn't even come close to answering.
Thank you for the update, Bridge. So the winds are picking up on us.
We got a sea cucumber over here.
Oh, nice.
Yeah.
Maybe.
This is in the sea pig group, isn't it?
It's something close to it. I want to say Amperima-like, but I'm sure somebody will correct me. But you can see the little feeding pads, the little feeding arms, and it's just kind of munching down on on some of the organic detritus that's present on the surface of the rock there.
So it's Elpidiidae, would you say? But I'm not sure which one. Trying to look it up really fast here.
You can see the surface of the animal is just gelatinous and very very soft.
Video come out. I'm going to try to come up above this so we're not behind that rock.
Oh, no, I think you're right. You said Amperima, right?
Yeah.
Yeah, okay, excuse me. Elpidiidae is the family, so that's it.
Elpidiidae.
Yeah. So possibly something like that. I'm just showing this.
Okay, come back in.
But for people who are watching who've perhaps never seen one before, note that the body is translucent, and what we're looking at is actually the animal's intestine, its gut. And so the sediment and the food that it's eating with the feeding tentacles on the left side, that's the head, are going into the mouth, and you can see that track through the intestine throughout the body. It'll poop those out later when it's done.
Boy, we're seeing some interesting stuff all of a sudden here on this dive. I mean, we had some stuff earlier, but we're seeing a lot of the same things, and now we're starting to see some different things. Oh, look at this. We got a black coral showing up right here. If we could get a zoom on this.
Is the current in this area higher? I noticed that we've seen a lot of current-based forms here, sponges and
I haven't
crinoids.
noticed very much at all. What do you think, co-pilot?
I don't think there's any current to speak of. If there was, this thing would be waving around.
But there may be at other times.
True.
Okay, video, let's try to do this floating.
Okay, I'm not gonna... This is definitely a black coral, an Antipitherian. I'm not sure I'm comfortable going any further than that. And Tina, let's see, I wonder if you're watching right now. We could use your help on this one. I know Scott's
up
probably...
to the left, almost finished here. That might be a small bad start for Chris Ma.
By the way, Ken Sulak looked up the Latin for pogon, and it had beard or hair, so it's referring to the attachment.
Oh,
serious. That's what it is. Thank you, Ken. That's great.
Yes. Thank you, Ken. Ken's a prominent ichthyologist, but as with all prominent or well-studied taxonomists, also knowledgeable in Latin.
This looks vaguely familiar. I'm hoping Tina is looking right now. I'm not sure she is, though, even though she's in the event log.
Try to get any details of this?
Yeah, if you could just...
All right, let me try to set a toe down
this,
here.
This is definitely a different Antipitherian than we have seen today.
All right, that should be stable. You can come in.
That's an amazing shot.
So for those of us who are, for those of you who are just tuning in, this is an Antipitherian. This is what's called a black coral. It doesn't appear immediately dark, and the reason for the name is that when you have the tissue removed, the skeleton underneath a lot of the tissue is actually black. I believe it's composed of chitin. Forms very interesting patterns under scanning electron microscope. So Tim Schenk from Woods Hole Oceanographic Institution asks if there are any associates. We're looking very closely, but I don't know that I see any right away.
Let's scan down.
I think this is possibly a Trisopathes, but I don't know. We've got one that's a question mark Trisopathes in the guide right now, the new version, which will come out shortly. I think that's about as good as we can go, a questionable Trisopathes, possibly in the family Cladopathidae.
I see no associates.
now that's an informative view right there, because you can see those little branches on the opposite side, which will help Tina and Scott come up with the correct ID for this. So yeah, thank you, pilot. Whenever video's ready.
All right, video. We have a swimming cucumber.
Ooh. Oh, where was that?
I don't know if I can get that. I'll try.
Ooh. Oh, there he is. I see him.
Was that the same Amperama we were just looking at?
It might be the same individual.
Yeah.
Yes. Came from that direction.
Thank you.
I'll zoom on this swimming.
So for those of you who are just tuning in, this is a sea cucumber, the same kind of animal that you might be familiar with on the surface as kind of a big worm-like echinoderm. They swim on the bottom. They move along on the bottom, eating organic junk out of the mud and the sand. But this type, especially in the deep sea, these are swimming, and so what you're seeing is the hood undulating. You're seeing the feet, the feeding arms, sort of spreading out as it's moving along. Such a graceful animal. Some of the weirdest animals I know are swimming sea cucumbers. And it's very nicely shot against this basalt background, so thank you. This is what's called a facultative benthopelagic swimmer. That is to say that this is a kind of sea cucumber which lives partly on the bottom, but every once in a while it swims to relocate itself to find better food. Sometimes they will sort of land and feed for a while, and then they'll sort of drop their load. They'll deposit some poop as they begin swimming up into the water column, so they can find a new place to find food.
A little chuckling there on your description. Apologize. Okay, moving on.
I'm talking about the defecation coming from the intestine.
I'm aware of it.
But they don't swim, they're not pelagic, they don't swim in the water column perpetually. That is something that we've seen before in another kind of sea cucumber called Pelagothuria, which is one of the only sea cucumbers that swims in the water column as a primary means of living. These do a little bit of both.
So we're having a bit of a pilot change-out here. That's why we're paused for a moment. That was a good opportunity there to take care of this, and then we'll be on the move momentarily. Trying to see who just came in. I suspect it's Dan and pilot. No, it's not Dan. It's Chris. Okay.
Oh, this is going to get complicated.
Oh,
quickly.
no, this isn't going to complicate. This is like a takeover. The Chris bunch now has taken over control of the control room.
Chris shift.
That's right.
Yeah.
We rule.
The CCC, the Chris Control Center.
That's it.
Nice.
I've got Don, I think, in the co-pilot position, if I'm not mistaken. And Josh is still there as the nav position.
Sean, he's debriefing, briefing Sean, Kennison will be in
How
shortly.
did Sean kneel down that small where I didn't even see him back there? I mean, the guy's huge. He's big, muscular guy.
He's nimble.
He's nimble. Wow. Okay, we've got a sponge here that would be worth taking a look at, I think, because it's the first one of these that we've seen all day.
Copy that, white?
Yeah, the white one. Thank you, Chris.
Sure. We're going to need to get a little ship move in if we want to get there. Sirius has a good head for that.
We were moving just due north. Do you want to keep going that way? It looks...
I'd say maybe 010.
010, copy. Do you want to move 20 meters, or would you like more?
So we got a question about how much bottom time we have left. Chris or Sean, would one of you be able to answer that question?
Yeah, well, actually, just give me 1 second to catch up real quick.
Sure thing. Yeah, thank you. Thank you, Nev.
It's Annie over here. Oh, he's back there.
Tim Schenk, who's watching right now. And I'm just getting caught up on event logs. We were watching all the commotion there, I see.
So, Chris?
Yes.
I think the other thing we have to consider today is the midwater transect, which begins at 4 o'clock.
Right.
So.
Correct. Right, they've got that factored in.
Yeah. Oh, no, I was just saying it's going to be about half an hour, 40 minutes.
Okay.
Oh, gotcha. Okay, I think we're almost stretched out there. How's the tether look? Good?
Yep, looks good.
Yeah, I can introduce a new group. I think Chris covered most of it, but we've got Sean Kennison at Navigator, Chris Ritter piloting, Don Liberatore at co-pilot, and Annie White video, and we all work for the Global Foundation for Ocean Exploration. And Roland Bryan is in the clipping chair, taking a back seat.
And so this is Chris Ma from the Smithsonian's National Museum of Natural History. I'm here with my co-biology lead, Chris Kelly from the Hawaiian Undersea Research Laboratory at the University of Hawaii, and Nicola Rodriguez, who is helping us with clipping and monitoring identifications.
Video. Start to get a zoom in here.
Seeing as species proceed.
Actually, can we get a shot with the lasers on?
Sure, go ahead, turn the lasers on.
So this is yet another type of glass sponge. So we're seeing a nice little variety of these today. This is a hard sponge. In other words, it's got sort of a fused together skeleton here. It's in the family Uncinetaridae, I'm pretty sure. And I believe it's in the genus Tritopleura. And those of you who follow the Okeanos expeditions, we've come across these fairly regularly just about everywhere we've gone at these depths. So there's a number of undescribed species in this genus. This is a little different than some of the ones, well, it's definitely different than the one we saw yesterday. So I'm just going to go to Tritopleura on this one.
So the two red dots that you're seeing are laser beams that give us a sense of scale for how large what we're looking at is. They're separated by 10 centimeters. So when you consider that relative to the size of the sponge, one realizes that this is quite a big sponge.
Let's come up on this. Low bear.
We used to call these leaf sponges because they kind of look like leaves sticking up out of the bottom. And they detach very easily. So
really? Interesting.
We found that out sort of accidentally from the submersible one year. When kind of just wanted to take a little piece of it and could have just grabbed a hold of it and had the whole colony in our claw.
Do you want to do these little white bits on the? Are those snails? I don't know what
they are.
Yeah, let's do it. Let's do a zoom there.
All right.
Amphipods.
That's full zoom.
I'm pretty sure. I'll see if Tim is watching right now and he could confirm.
Is that full, you said?
Yeah, that's full.
Copy.
That's just sort of noting that we saw a bunch of these chrysogorges that were coming through too, sort of a little field of them. Roger, Nav, go ahead. Roger that, 30 minutes of bottle time, thank you.
All right, video's clear whenever.
Did you copy? We have about 30 minutes.
Those of you on shore didn't hear that. We've got 30 minutes left. Sean, can you tell us about how far we are from our endpoint?
Okay, let's come wide.
All right.
Video. Do you want to do a whole specimen?
Just something like this is good.
Okay.
Lasers off?
Sure.
All right, I think we're good.
Roger that, 180, thank you. Navigator.
Come on out. So we're full wide.
Great, thank you.
Rogers. It's got a little reflection to it, doesn't it?
Yeah, it does.
So Tim, he had a hard time really seeing them very well where he's at. So he wasn't really sure whether they're amphipods or snails or large forams.
Yeah, I thought snails.
Thought snails?
Yeah.
Okay, alrighty.
How far are we in that ship move?
I just got word from Bridge it just ended.
Copy.
So there was about 5 minutes of delay earlier today, so you might just start to feel it now.
Okay. Well, let's go ahead and get another one in.
Copy. Do you want to keep moving in 20-meter steps?
Ooh, I think we can do more than that.
All right.
30 sounds good.
30 and keep 0, 10, 0.
Does that move towards your end point? That's about upslope, yeah. Maybe
yeah.
a little bit more. 015 maybe?
Yeah, sure.
015?
Sure.
Copy that. I'll put it in.
Just looking between my toes here. Annie, you want to try to get whatever is swimming here?
Yeah.
If it is something.
Looks like it's something.
Yeah.
What's that?
That could be a Ketognath, but I'm not sure. Maybe
That's
not.
Oh,
Whoa! Oh, yeah, it is.
This is an isopod. This is a relative
pushing,
of the little pill bugs you see in your
Did you say keep pushing in?
Yeah, if you
yard.
If you can,
But
I'm just going to
they
creep
have
in
really
on it.
bizarre adaptations.
That's not quite full
that,
zoom.
Okay,
they
let's
give,
just hold there
it
for
gives
now.
them the
Yeah.
ability to swim. These are really long legs that they're using. These are found in the midwater, in the sort of three-dimensional water space that occurs above the bottom of the ocean. It's delightful. You can see it using its other legs swimming upwards through the water column. But they look almost like spiders. Oh, that's beautiful. And they'll see, from what I've seen before, they do this. They have the same behavior. They kind of climb and swim, and then they stop and they break open their legs and they float for a while.
So I was close. I thought it was a Ketognath first.
Oh yeah, well it was a
was
small linear thing.
Sort of joking. God, I was off on that one, wasn't I?
Oh, I was
Thank
being
goodness he
nice.
opened his arms up. No, you don't have to be nice to me. I haven't been nice to you. I tried to get you on the Sea Star. So. No
All right, let's let that go.
All right. Coming full wide.
Okay, isopod. Thank you.
And we're wide.
Copy. Hard to track that guy.
This is a hard game we play. Trying to ID stuff from video. It's can be very, very challenging, or is all the time.
Is that something else, water column?
Yeah, there is something there.
Oh, Roland may have spoken the magic words. Possibly a jellyfish.
Okay, go ahead from here. Just creep in here.
Or
It's a crinoid.
Oh, that's interesting.
Yeah, he's drifting.
So this is a feather star. I had mentioned earlier that sometimes when they're provoked, they can launch themselves into the water and swim. Usually,
That's a
though, this
partial
is in
zoom.
response
Okay,
to some kind
go
of
ahead and give some more.
defense.
Pretty
But this one is just sort of hanging in the water
less
column.
mobile
That's
than
kind of unusual.
previous subject.
Unless at some point it got motivated into the water by our actions, or perhaps those of a fish, or perhaps even those of their enemy, the sea urchin. Huh.
That's pretty high. We're 2 meters up. So
yeah. I mean, it's
not settling out on the bottom either. He's just kind of hanging in the same.
I mean, dare I say the words pelagic crinoid? I'm sure my colleagues will be horrified at my making unsubstantiated conclusions. Wow, pretty, whatever it's doing.
All right.
Thank you.
Yeah, thank you.
That's way out there.
We're full wide.
Copy. It's coming back down now. Pretty low. Anything out there, Annie?
I think just the corals and sponges.
Kind of hard to see if there's anything that we haven't already zoomed in on.
Yeah.
Yeah, I think that was a Chrysogorgia. There's a coral to the left there, but I think that was something that
we've got a lot
seen
of
before.
Yeah, this is the Chrysogorgia verda, I think, the stocked one. Doesn't mean we can't look at some things again. They look a little bit different. This is, I think, the little grouping that Les was talking about, little grouping of the Chrysogorgids.
Ah, okay. I think that might be a shiny.
All right, pilots, that last move just finished.
The base
maybe.
Base system.
Yeah.
I'd say
keep on going.
Just keep going. Yeah.
Excellent. I can just keep doing 30 meter moves.
Yeah, let's do that.
At 15
Is that
degrees.
a sea star or is that a coral on the?
I'm thinking more coral or something.
Yeah.
Okay. Or
15
criner,
is good.
I don't know.
Yeah, center looks good. I'll put it in.
Yeah, you're right. I think it's a small,
It looks
possibly dead
frondulose.
coral. Yeah.
Oh, look at that. Looks like a big. Is that? No, that's a sand patch.
Yeah.
Excuse me.
We're getting late in the afternoon IDs, I think. That's what happened. I think that's what. Another bolosoma in the distance, and another one of the other glass sponges, and probably the genus Polyopicon just below that to the right. These corals right here in front are Chrysogorgids primarily.
Oh, is that an Iridogorgia up there?
Where is it? Where do you see it?
Center there?
No, that's another one of these Chrysogorgids.
Oh, that's another Chrysogorgia.
It's just a different orientation.
Okay.
There is the crinoid sitting on the top like a little crown. Maybe we could take a look at that.
Sure.
Oh, this is that funny thing with the.
You can follow me around.
Dark tips. Yeah.
Metris something or other.
Right, the Xenometrid.
Yeah, the Xenometrid, but whatever. Chuck Messing's out there. We had a great easy name, Cerametra, until he told us we were wrong. Now we got to memorize a much harder name.
Wow, I love the way that just sort of, it's like a deep sea Christmas tree. Sathyrometra.
Sathyrometra.
Yeah.
Okay, video. Go ahead in.
All right.
Oh, that's lovely.
Yeah, that's a beauty.
Yeah, so the characteristic of this one are those really tapering arm tips, as well as the distinctive color.
You're free to zoom however you'd like there.
So that's Chrysogorgia?
Yeah, that's a Chrysogorgia, and Les will probably, he's still watching. He might be able to help us out on the species. Oops, I haven't been monitoring event log for a while. Sorry.
Hey, Chris.
Yep.
Yeah, there's an ophiuroid in the background on that other. Is that Chrysogorgia?
Yeah.
Thank you.
Boy, I haven't looked at the event log for a few minutes. Excuse me
take
here.
a little
Let's
more
see
zoom.
if we got some
Sure.
requests.
You want to go for purple base?
Yeah.
Okay.
Les is asking whether I've been calling the cylindrical Chryso C. geniculata. I did earlier, Les. I think I've just been calling them Chrysogorgia for a while when we've seen them.
Sorry. Yeah. So these kinds of views are really great for people who study crinoids, because a lot of the ways that people who study them identify them is through a very complex observational study of the way that these little skeletal pieces on their arms fuse together. They're called syzygies. It's where they fuse to, where sometimes the individual plates actually merge together, as well as some of the other details on the what's called the pinnules, the projections that come off the arms. Skeletal morphology is really important in polychinoderms that have a skeleton, and so these are very intricate, sort of like living three-dimensional puzzles. And it's really wonderful to see them so close and alive like this, especially with the colors.
Let me know when you're all set.
We're all done. Let's move on.
We're all done, and we had a request to look at that brittle star that Chris Ma was talking about a moment ago that's behind this as you zoom out. I don't know if that's possible, but there he is.
We could take a quick look.
Yeah, maybe a quick look at him.
This is only partially out.
Puppied off a little. Yeah. Okay. Let's just try to get it from here.
Okay.
Okay, go ahead.
And that's full.
Copy.
So this is a member of the ubiquitous deep sea group, the Okeanos. It's interesting because I wonder if it's just about to jump off the way that some of the others seem to do. Every time we come across one of these, they seem to be very active, or at least distressed by whatever it is we're bringing to the scene. But I don't recognize the genus right off the top of my head, but some of the folds across the disc suggest Ophioplenthaka. I can spell it for you later. You've already annotated it.
Yeah, and also Tim was pointing out that there's a squat lobster there to the right of it, upper right of it. You can see him right here.
Yeah.
And possibly a Europtychid. Europtychus.
Like a squat lobster of some kind.
Yeah.
And so we were talking earlier about how these different corals actually provide habitat for other animals, and it's frequent to encounter these with squat lobsters and with brittle stars living among the branches.
All right.
We got to run here, unfortunately.
We're full wide.
Copy.
Thank you, pilot.
Sure. We got to come back to our heading here. Came way off.
Okay. Great. Thank you, Chris.
Let me know when you're back on heading there, Don.
Yep.
We've got a larger branch coral coming up. See it on view. If we could get at least a snap zoom on that.
Yeah, we can get a better shot on this. It's more aligned with our head. So.
Okay, we're in there now.
Copy.
Straight now.
You'll find one of your evil beasts on it. It looks like one of these planar bamboos coming up, and we often see sea stars. And there's something red on the.
I don't know. I don't see any.
Is that it may have been a laser dot right at that moment.
So what Chris is talking about is we frequently find these bamboo corals And I would often refer to these bamboo corals as starfish food because there are several deep-sea goniasterid sea star species which prey on the tissue of these corals. This one, however, looks like the coral is mostly dead anyway, so there's a brittle star hanging on the tip there, but...
okay, let's fill frame.
Alright. Can we get the lasers on?
Sure, go ahead. Let me tilt up a little for you.
All right, Les, you're on, buddy. One of your bamboos is
lasers
coming up here.
Yeah,
Could use the help.
Oh, okay, yeah, they're just...
And you're going to wait until he zooms in. He's doing the colony shot
okay,
here.
Yeah,
and
go
then
ahead.
He's going to zoom.
Okay. That's partial.
Okay.
The branching is internodal, sort of.
That's full.
Kind of nodal, internodal-ish.
Yeah. Well, and let's head up to the right part of it where the brittle star was.
Well, let's see if we can ID the coral first, and then we'll get that brittle star. Let's give him a chance to look at the polyps here.
There were some hydrids off to the left side as well.
Trying to make it hard for Les. He's got to study the actual spicules on these things to have a chance. Let's see what he comes up with. I guess you call this nodal. Ooh.
So Les reports J-clade.
J-clade, close to Jasonisis. Okay, great.
Okay.
Now let's go see what that other... See, now these are friends of corals, these brittle stars. These will chomp down on them like your beasts do. No.
This is interesting because the surface that it's perched on, all of the tissue has been is absent.
So
another Okeacanthid. What?
Is he eating it, or is he just rubbing it off with his arms?
It wouldn't surprise me if it was either explanation. I mean, those spines are pretty
close on top of you.
Those spines can be pretty sharp, so it doesn't like facing us.
Yeah, that's okay. What's your camera at?
Oh, that'd be nice if it could actually turn around and we could see the mouth.
Okay. I think that should be stopped anyway.
If we could zoom in on the mouth, that would be the little orange spiny bit right on the underside.
Is that full video?
Yeah, this is full.
Any second he's getting ready.
Copy.
That's full zoom, watch lead.
Thank you. Oh well. Still, great shot of one of these. It doesn't like us. Brittle stars are so antisocial.
Look at that. He's waving.
Yeah, it's waving the other direction. The face is going. I love the way the spines are arranged in a single plane, though.
Yeah. But so, anybody? So Okeacanthid is your best.
Yeah.
Okay.
I think it might. It has the folds along the disc, so possibly Ophioplanthaca, but I'll have to double check.
Okay, and then he'd mentioned there were some hydroids maybe on the left side of the colony. I wonder if we could take a peek at that before we, and then we'll have completed our documentation of this colony.
And then video spied an anemone, we believe, past this to the right.
Oh, yeah. Okay.
It looked very Relicanthus-like.
Oh, sure. Yeah, let's go take a look at that.
Oh, yeah, this is very nice.
Yeah, these are hydroids, I guess.
We're looking back at you now.
Copy.
And what has it got on it? Some snails?
Yeah, I think they are.
How long has the ship been stopped? No.
Probably, I think about 3 minutes.
Okay.
So
you
we're
might
seeing
have a couple stacked
an array of
transits, so it might swing more than
sort
that.
of colonial animals covering the surface of this bamboo coral, in addition to several what it looks like snails.
Pretty good.
Okay.
Similar to the ones I think that we saw on that sponge.
All right, watch. So if we want to get a shot of the anemone, we're going to have to go here.
Let's get the anemone.
Okay.
Quite wide.
The anemone.
We're full wide.
Copy.
Undoubtedly deposed down here because there was an anemone of the people.
All right, so Annie, where's this anemone? Let's see if video is as sharp as they think they are. Let's see where it is.
Yeah, it's right center screen right now, just below the lasers.
Ooh, she's pretty sharp. She's spotted it. Way to go, Annie.
Okay. We've got about 30 seconds or so.
Okay.
So let's make it happen. Okay, go ahead and zoom.
So is there any chance that we could consider collecting this? The reason I'm suggesting this is on the collection list.
Just go ahead and back the ship up a
This is a really weird serianthid that lacks a tube, and it lives on rocks. And this has been a curiosity for years. We don't know what it is. So this is not relicanthus. If you actually look inside the mouth, you'll see the oral tentacles around there.
I see it. But Pilot, did you just say we had only a few seconds left?
Stand by one, Watchley. Oh. We're backing the ship up right now, so we'll have to. Let's drop a target here. And
is that okay on shore? This is something we've seen before. Those of you who've been following.
Should I come wide?
And
yes, please.
We just really have no clue.
Or full wide.
Except that Tina has identified this as a serianthid, and that's the best that we can come up with.
Okay, you can start to come up a little bit, yeah.
Yep.
So, watch lead, if we want to collect that, what we're going to do is I'm going to push ahead just a little bit, make sure we're safe. We got eyes on the tether, and while we're moving the ship back, we'll backtrack to it.
Lovely. Okay,
does that sound like a good plan?
Yeah, it sounds like a plan.
Okay.
Splendid. Thank you.
This would be a great final biologic to get a hold of this thing and finally answer what the heck this thing is.
Good.
And that'll likely be the end of the dive, I think.
Okay, well, what a way
At least
to
on
finish.
the bottom.
Yeah, exactly.
Thank you.
See, this is
a beautiful color.
It's a beautiful color. That's as far as anybody could go on this thing. Seriantheria is the best that
You think we should back up more than 15? What do you think?
Probably.
Yeah.
Move it out to 20 or 25.
So it's a real problem when you call something to an enemy and it doesn't have a tube.
Yeah, if you don't mind.
I have that problem all the time, Chris.
Did you note the depth right where you were sitting on that object?
I didn't, but he dropped a target. So.
Great, guys. We do have a few. There is some support in the event log to give it
Depth
a go.
has 2413.
2413.
Which does not look right.
I think we can find it.
Okay.
Okay. And from what they told me before, you got the starboard box.
This thing go into
port side's full. Starboard's got one open.
Copy that.
What do you
don't know whether it's the inner or outer. So.
Well,
is it the same
it's
as an enemy?
not unusual to take us.
We would obviously get something for DNA on this.
Yeah,
thing for sure.
to take a clipping for DNA, but in a lot of times the bodies go into something more like formalin.
Okay,
So, in preparation for that collection, Watchleys, how much of the animal are we talking?
The whole thing, if we have any chance of identifying if that's possible.
So closest to the rock that we can get?
Yeah.
Copy.
Hopefully it'll pop right off the rock intact, if we're lucky.
Sure.
Yeah, no.
We got rocks for them.
No, he's just saying that a lot of times you can get the animal in perfect condition if it's on a rock where you could grab the rock. But it's not going to happen this time, I don't think. I'll just throw this out to shoreside. We are going to go ahead and put this in formalin, 10% formalin, unless somebody knows more than we do about how to preserve something that's been identified as a
just don't change it so that I can, as a reference, you know what I mean, going back.
Yep.
Know that anemones really need histological examination of the mesenteries in order to identify
and we know it's
them.
in front of that other coral.
So I'm not aware of seriantherians having hard parts that would be degraded by formaldehyde, right?
No. I think that's completely appropriate.
Okay. This one beast is going to use up our entire formalin stash for the cruise.
Are you backing up?
Let's see how big it is.
I'm backing up.
I think it's going to be pretty big.
It might fold in on itself.
Yeah, that's true. Interesting to see how it responds to when it's touched. You know, most Syriantherians were tracked into a tube, but this guy doesn't have anywhere to retract.
It could still withdraw into itself, like like a sea anemone would.
I see.
We're just slowly working our way back. Ship's taking a little while to catch up, so.
Okay, thank you, pilot.
Sure.
Yeah, it looks like ship's just about 25 meters back now.
Over on the left.
Okay.
Yep. Coming down a little bit.
Sure.
All right, that looks familiar over there, the little piece on the bottom.
Yeah, it does, doesn't it? You want to go ahead and get me spun up on hydraulic?
Sure. You're up.
That skeleton looks like a different type of sponge, so I'm wondering about the idea, the one that I threw out as Phytoplura, because we're seeing the backside, we couldn't really see the front side. This skeleton looks like it's something in the genus Aspidoscopulia. I wonder if that's it.
I wonder why they're all dead.
They just natural, of
fall
course,
over,
they get
you think?
old. Yeah. There it is. All right.
Okay. I'm right above that coral, so I'm gonna.
Maybe about 10, 15 centimeters across or so, maybe something like that.
Yeah.
Yeah. Now you can see the oral tentacles, that little dark center of it, because he's kind of spread out a little bit.
There's the lasers coming across it now. There you go.
Oh, perfect. So
yeah, I know we were back.
The disc itself is about 10 centimeters or so, and then the arms take it out double that.
Oh yeah, so it's a big animal.
Yeah. Hope you're right. I hope it closes up on itself.
Well, it does seem to be on a rocky substrate, so as you said, there's no tube. Go ahead.
Understood. Thank you very much.
No problem. Thank you.
Pilot?
Go ahead.
We should probably get a close-up, because we didn't get one earlier.
Copy. Okay, go ahead.
All right. That's partial.
Oh, terrific.
Roger.
Nice colony shot prior to the collection. So he is on a stalk under those tentacles. It's probably sticking up at least a few centimeters above the rock.
Yeah, you go ahead. Zoom if you need to.
Wow.
Yeah.
Do you feel comfortable changing that camera?
Yep.
co-pilot?
Sure.
To Mini-Zeus, I mean?
Yeah, no problem. You ready?
Yeah. Good. So you just keep an eye on that in the other camera there.
HD2, there we go. You want me to take Mini-Zeus, or you got it?
Sure. Go ahead. Okay, Army's alive.
And video is clear on the close-up whenever you want wide.
Copy that. Yeah, you can come wide.
Okay. That's full wide.
Copy.
Pilot, just a heads up. Port box, bio box is full.
Copy.
Inner and outer, so you might have to go starboard with us.
Yep.
It's going to be the moment of truth.
Roger that.
But just to see how it responds.
Give me a partial zoom out there, Annie.
Yep. You want more than that?
No, that's good.
Okay.
It is detecting. Whoa! What is it doing?
Should I
look at this?
Let me know if you want me to zoom out.
Okay, zoom out. Zoom out.
Well, that was unexpected.
Or full wide.
Yeah.
Do you want me to try to still collect it?
Yeah, absolutely.
I'm going to open the box up.
This is actually not
goodness.
unexpected because you have sea anemones that escape sea stars
tilt up my camera?
by jumping up and swimming away.
You might be able to push it into the box even rather than...
Come down on Mini-Zeus.
Coming down.
Flying right into us.
Yeah.
Well,
where did it go there?
It's
straight
back. Just
you get it on
seeing
text all
that
the time?
reaction is amazing, even if we can't.
Huh. Wow. I would not have.
Can you brighten up Mini-Zeus?
Yeah, you
Any
should
chance?
see
I mean, I've seen other sea anemones do that before, but I guess I wasn't expecting it. That's weird.
I see it.
Wait, you think it's in the...
It went under Zeus.
How do I get...
We have reactions from, well, that was a surprise, wow.
If you just back up, it might come
in the event log.
back out.
Jeez.
Hey, pilots, that is
That
all the
caught
time
everybody
we have.
off guard.
Just to make the transects, I'm going to have to go ahead. Watch, leads, this is Nav.
Roger, now go ahead.
Hey, yeah, it's time to come off bottom. So unfortunately, unless you want to take time out of transects, we're going to have to start to set up.
It may have got stuck in the water.
I understand.
train
No,
going
let's
up to the
keep to the
thrusters.
schedule. I think next time we'll be prepared for that reaction, and
Yes.
we do see them. So
Absolutely.
that's fine. Thanks very much for trying, and thanks, Andy, for spotting that.
Unless it's somehow attached itself to the ROV.
All right, pilots.
Yeah.
You got it.
Copy that.
All right.
Yeah,
Thank
we're
you
gonna,
so
we're
much.
gonna have to
That's-
skip collecting it, guys.
I think that was a lesson in and of itself.
Stow the arm.
Yeah.
Yeah, I hope that makes a highlights clip for this dive.
Oh.
That was extraordinary.
I might be able to clip one or two pieces out for you.
Plus an appropriately amusing soundtrack.
Okay.
So
Well, we saw a very effective escape mechanism there. Got away from a 10-ton
Oh, yeah.
Beast.
Okay, hydraulics are off.
Okay.
Yeah, I didn't want to just squeeze it in half.
It down, yeah.
And I didn't know if...
So
no, that was fine. We'll have another shot at those. Yeah. We see
at
them
the,
every
at the time.
Yeah. So it may have gone underneath.
So we're wrapping up here then, as far as the bottoms.
Yeah, we're going to do the midwater. We're not going to cheat the midwater.
Yeah.
Okay.
Oh, no, that's fine. But so we're wrapping up from the bottom part of this. In about an hour, we're going to conduct a midwater survey and look at pelagic animals. So rather than looking at animals which live here on the bottom
we're going up
of the
to
seafloor, we'll be
meters,
looking at those which live in
yes,
the water
okay.
column,
800
jellyfish,
meters.
squids, and other animals that
and we want to do
have a primarily midwater habitat.
Typical.
Today we saw a very amazing
recovery,
up
to 800
aspect of sea anemone behavior we wouldn't normally have seen. We saw lots of glass sponges. We saw sea cucumbers. We saw a number of very interesting corals, collected some potentially new species, and saw some very nice deep sea bottom fauna. This is Chris Ma from the Smithsonian's National Museum of Natural History, co-biology lead Chris Kelly at the University of Hawaii. I'll be back in a little while to help with the midwater transect, and we'll see you all in a little while then. Thank you.
Yeah, thank you. And then what about the dive, post-dive, and planning? Kelly's coming in right now. Yep. Yeah, why don't we do that? Those of you who'd like to participate in the dive site planning, we're going to just get set up. Give us just a couple of minutes for Mashkor Malik to bring up the Fleeter Mouse display on feed 3, and we'll roll right into it. Thank everybody for their participation today, whether you hang on or not to the planning session.
Starboard.
Okay.
And line up with the ship.